Good morning, sir.
Yeah, good morning.
Good morning, sir. Welcome.
Thank you. Yeah, thank you.
We'll
Thank you.
start, sir.
All right.
Good morning. Sir,
Good morning, Prakash. Everything is set?
Yes, sir. Yes, sir.
Sudhindra, hege ella ready?
Yes, sir.
Very good morning to all the dignitaries. We are currently live on YouTube, and I hope that I'm audible and visible to all the dignitaries right now.
Yeah.
Already live.
So, a very good morning to everyone. I'm Dr. Siddharth Pandey, a senior resident in the Department of Internal Medicine, and I welcome you all to the CME titled Preparedness for Disease Outbreak, being conducted by the Department of General Medicine, SHEMA, Nitte Deemed to be University, in association with API Dakshin Kannada. Charles Darwin said, "It's not the strongest of the species that survives, nor the most intelligent that survives. It is the one that is the most adaptable to change." In this changing world of modern medicine, we've been bombarded recently with a pandemic, which has changed the way we practice medicine and even how we go about our daily lives. This CME is a humble attempt by the Department of General Medicine to abreast ourselves with the latest developments so as to enable us to adapt to possible disease outbreaks in the future. God's first line of defense and offense for every situation is a prayer. Now let us begin by invoking the blessings of the Almighty. I now invite Ms. Megha and Ms. Kripa, final year MBBS students from SHEMA, to recite a prayer. Megha and Kripa, please.
अक्षय भक्ति संस्थितये पूर्ण ब्रह्मा रूपिणं परिपूर्णदयारस पूर्णिदलोचनं पार्थमहाभक्त भावितमच्युतं तीर्थपदं महीलाकविदायकं वैकुण्ठादीपं विबुधगणार्चितं श्रीललिताम्बिका स्तोत्रं परं पूर्णत्रयीशम् आश्रये अक्षय भक्ति संस्थितये.
Thank you, Megha and Kripa, for the divine recital. I now request Dr. Sudhindra Rao M., the head of Department Internal Medicine, K.S.HEGDE Medical Academy, to formally welcome the delegates to the program. Sir, please.
Thank you, Siddharth. Thank you, Siddharth. So, good morning to everyone. I'm very happy to give the welcome address on behalf of the Department of Medicine and welcome everyone for this program. Department of Medicine in K.S.HEGDE Medical Academy, which is a constituent college of Nitte University, has been conducting annual CMEs for more than 5 to 6 years now. And every year we would do the CMEs on a theme. And this year we have chosen the theme of preparedness for pandemic. And this is the first time we are going on a online come a hybrid CME, online come offline, which is a hybrid CME, where our institutional members are here with us, and the speakers and the delegates from across the places are coming and visiting us on YouTube and watching this. So we have been hit badly by the pandemic, and there are other outbreaks which are happening every now and then, creating worries in the mind of people, and that's why we have taken up this CME. First of all, I would like to welcome our VC, Honorable Professor Dr. Satish Kumar Bhandari, who has been the inspiration for us to conduct programs like this every year, and he has been a motivational force to us. I also welcome Dr. Prakash P.S., who is our Dean, and he is the of all the support for us in all our endeavors, and he is our own previous HOD, and sir has been very supportive to us. I also welcome Dr. K.S.HEGDE, President of API DK chapter, to this forum, and the API has been of lot of support for us in conducting this program. Like yesterday we conducted the paper and poster presentation where API members actively helped us. I also welcome all our speakers. So this being a hybrid CME, we are lucky, or we can call it as a blessing in disguise, to get world-class speakers from across different states who will enlighten us on the different aspects of the our preparedness for the pandemics or outbreaks. I also welcome all our delegates who will be watching us on the YouTube today, and I request everyone to get the most benefit out of this program and from the expertise of the speakers. I also welcome all our institutional members, our students, and everyone for this program. Thank you, Siddharth.
Thank you, sir. Association of Physicians of India is a professional body of consultant physicians with postgraduate qualifications in different specialties. It was formed in 1944, and the current membership exceeds 19,500 doctors, which is again an ever-growing number. The API has numerous publications under its belt, and the Indian medical fraternity owes it most to them to put modern medical literature in an Indian context. Today's CME is being conducted by our department in association with API. I now invite Dr. K.S.HEGDE, President API Dakshin Kannada chapter, to welcome the delegates. Sir, please.
Yeah. Thank you, Dr. Siddharth. Good morning and a warm welcome to everyone. The Nitte University is a prestigious one, and under the great leadership of Sri Vinay Hegde, Sri Vishal Hegde, and the Vice Chancellor, Dr. Satish Kumar Bhandari. Now, disease outbreaks can be devastating one, like the one which we are experiencing presently. So in this time, it is very apt that we are prepared for such disease outbreaks. So today's CME is a great initiative in this regard, and it is done by the great institution K.S.HEGDE Medical Academy under the guidance of Dr. P.S. Prakash, the Dean, and Dr. Sudhindra Rao, Professor and HOD of Medicine in Shema, and all their team. So we in the API Dakshina Kannada are very pleased and privileged to be partners in this CME. We are happy, and a warm welcome again to all the dignitaries, the speakers, and the participants. May this CME enlighten all of us. Thank you.
Thank you, sir, for your kind words. I now invite Professor Dr. Satish Kumar Bhandari, Vice Chancellor, Nitte Deemed to be University, to deliver the inaugural address and formally commence the program. Sir, please.
Very good morning to each one of you who are participating in this very unique webinar conducted by the very prestigious department of the K.S.HEGDE Medical Academy, Internal Medicine Department. The President of the API Dakshina Kannada District, Dr. K.S.HEGDE, the Dean of the Medical School, our own, the former head of the Department of Medicine, Dr. P.S. Prakash, head of the Department of Internal Medicine, Dr. Sudhindra, and his team. Very distinguished speakers, Dr. Yamuna Devi, the consultant in infectious control, Apollo Hospital, Dr. V. Ravi, popularly called Virology Ravi, the nodal officer for genomic sequencing, SARS-CoV-2, Government of Karnataka, Dr. Nitin Gupta, the consultant in the Department of Infectious Disease, KMC Manipal, Dr. Anusha Rohit, our own alumnus from K.S.HEGDE Medical Academy, presently the head of the Department of Microbiology, Madras Medical Mission, Chennai, and all distinguished faculty from the Faculty of Medicine and the delegates who are participating in this webinar. I deem it my privilege to inaugurate this unique webinar conducted by the Department of Internal Medicine, Kshema, in collaboration with the API Mangalore chapter, this Dakshina Kannada chapter rather, on an interesting topic of preparedness for disease outbreak. I should congratulate the Dr. Prakash, the Dean of K.S.HEGDE Medical Academy, for guiding the Department of Medicine, and also head of the Department of Medicine, Dr. Sudhindra, for selecting this topic, a slight deviation from the usual topics of COVID. They have taken a wide area of preparedness for disease outbreak. Congratulate the Department of Internal Medicine and the whole team who have worked for preparing for this webinar. As you all know, we are going through an unprecedented healthcare crisis globally due to COVID-19 pandemic. Already more than 200 million people have become sick worldwide, and more than 4.5 million people have died due to COVID-19. And many survivors are experiencing a long haul of symptoms still. Despite advances in the vaccination and clinical management of patients with COVID-19, the end of the worst public health crisis of the century is not nearly in sight yet. COVID-19 has impacted many different aspects of society, from politics, economics, to the psychological well-being of citizens, and the list continues to grow as the spread of corona persists. While it is impossible to fathom the way COVID-19 will change our usual way of life, there are many prevailing concerns that the community currently faces. Apart from the impact of this disease on the physical health, we don't know the answers for many long-term effects of such diseases and outbreaks which may come in the future on the mental and spiritual health of the people. Scientists have predicted that prolonged lockdown and closure of schools can cause long-term effects on learning ability and even communication skills of the particularly children. We have got a lot of concerns and we had challenges during this pandemic. We had a lot of questions to be answered. How do we enhance the collective resilience of community during this pandemic? How do we cope with the mental health issues during pandemic? How do we deal with the bereavement of our kith and kin during this pandemic? How can we support healthcare workers and emergency responders during this pandemic? How to provide universal health care to such a large number of people, such a huge country? How to create sustainable, cost-effective infrastructure for a large, populous country like ours? Therefore, there is an urgent need to enhance our understanding and the level of preparedness against COVID-19 in situations like pandemic situations, not only COVID, the conditions are going to emerge in the future. The COVID-19 pandemic has opened an window of opportunity for rethinking the way countries prepare their public health sections. On one hand, there is a sense of scientific accomplishment and that the global research community has collectively risen to the challenge imposed by the pandemic. In no other time in history of science has so much information been generated, shared, and deployed so quickly to address the threat to the humanity. On the other hand, is the realization that no amount of research and the scientific development can compensate for ineffective leadership and the lack of coordination between the government and international cooperation between the countries. Experience from the recent past has repeatedly shown that the resources, funding, and political capital tend to taper off as the outbreak recedes. Public memory is very short. We forget that we had such a pandemic and we have passed through such a grave situation. The true impact of COVID-19, however, in both health terms and economic terms, is likely to outlast the duration of the active phase of the pandemic, opening a window of opportunity for enduring changes while the virus still prevails in the minds. The window of opportunity must not be wasted. There's a clear sense of preparedness for the next pandemic. It will require a deep dive into those future failures, understanding what went wrong and what worked for improving the architecture of health security governance. The world is still pretty much in the midst of crisis, and one might argue that it's too early to start talking about preparing for the future pandemic. We disagree. The Department of Medicine has taken a right step now in the right direction to talk about the future and about the changes that need to happen to improve the global capacity to respond to health crises of this magnitude. I'm confident that our very eminent speakers, very distinguished speakers from various parts of this world here today will enlighten us all over, and also the participating delegates will be benefited by this webinar. The topics selected are very, very interesting and thought-provoking and is very, very contemporary. I hope all the participants will have very interactive and fruitful webinar. Thank you very much for giving me the opportunity for participating in this inaugural function. Stay safe, stay healthy. May God bless you all. Thank you very much.
Thank you, sir, for your inspiring words. Well, as John C. Maxwell says, a leader is one who knows the way, goes the way, and shows the way. In keeping with these words, I now invite Professor Dr. P.S. Prakash, Dean, K.S.HEGDE Medical Academy, a constituent of Nitte deemed to be university, to say a few words. Sir, please.
Good morning, Honorable Vice Chancellor, Dr. Satish Bhandari, Dr. Sudhin Rao, HOD of Medicine, Dr. Ganesh, President of API-DK chapter, today's resource persons, Dr. Yamuna Devi, Dr. Ravi, Dr. Nitin Gupta, of course, our own alumnus, Dr. Anusha, my departmental colleagues, API colleagues, and my dear postgraduates. It's indeed a great pleasure and my privilege to give this address on the occasion of CME, Preparedness for Disease Outbreaks, jointly organized by Department of Medicine and API-DK chapter. At the outset, let me congratulate the organizers for having aptly chosen as a subject which has been a very relevant topic for this day. Ladies and gentlemen, all of us are aware what we have been going through in the last 1.5 years as healthcare providers. Not only that, it has been no less taxing on the authorities, be it frontline workers, district and state administrators, economy, livelihood, and of course, I feel personally, worst of all, my medical education, because I see students who have missed a lot of things during this pandemic. In the past, of course, this disease has no parallel in the history except for all of us go back to 1918, the Spanish flu pandemic. Otherwise, there were a few diseases which threatened to become an outbreak. That was the plague of 1994, H1N1, SARS-CoV-1, Nipah, etc. Luckily, it did not, we were able to contain it well. Now, considering all these aspects, to prevent and contain outbreaks, all countries and international organizations should be adequately prepared to detect outbreaks early and respond promptly and effectively. In fact, if you look into the disease outbreak cycle, the first one is preparedness, next is detect, respond, learn, and again the cycle repeats. However, in most countries, there is underinvestment in outbreak preparedness and over-reliance on the outbreak response, which we have been doing. Of course, we did not have the experience, neither the medical community nor the administrators. So remember, outbreak response is a costly affair compared to outbreak preparedness. Science has advanced significantly and new technologies have come up. We are still not able to prevent the new outbreaks. Dear colleagues, outbreak preparedness is an essential component of outbreak response cycle, providing health security to all the population or its citizens. Preparing for outbreak needs careful planning, adequate and optimal use of resources, and when done is highly effective in saving human lives and of course its impact on the economic aspects. On this occasion, the Department of Medicine and API-DK chapter, I again congratulate the topic chosen, and we have a galaxy of subject experts who shall be addressing you on various aspects of preparedness for disease outbreaks. If I look into the chapters chosen, one seems to be a bacteriology, one seems to be a virology, and another seems to be a mycology. And I think finally the microbiologist, a senior microbiologist from my own student, is going to summarize all of these things. Hope all of you will enjoy this CME and become that much more knowledgeable at the end of this CME. I wished it all the success. I thank the Department of Medicine and the API-DK chapter for organizing this CME. Thanks one and all the best. Thank you.
Thank you, sir. Without further ado, we now begin our CME formally. At the outset, I'd like to brief you about the program today. I'd like to tell you that our hybrid CME actually started yesterday, and our department successfully conducted online research paper and poster presentations, wherein 7 research papers and 8 posters were presented both in the competitive and the non-competitive categories. Today, we have for you a series of 4 lectures by experts in their respective fields, each of whom will be formally introduced to us by the members of our faculty. The lectures that we are presenting as of now are being conducted on Zoom calls, which will be relayed to the participating delegates via a YouTube live stream. Since there is a delay of around 2 to 3 minutes between the Zoom calls and the relay on YouTube, as we would experience in a televised news telecast, it is not possible to entertain direct questions to the speakers. However, any genuine queries of the participating delegates can be put forth in the YouTube comment section along with their names, and the same will be relayed to the speakers at the end of the presentation if time permits. So, an American writer, H.P. Lovecraft, says, "The oldest and strongest emotion of mankind is fear, and the oldest and strongest fear is the fear of the unknown." Our first presentation is an approach to unknown outbreaks by Dr. Yamuna Devi R. I now invite Dr. Supriya P.S., Associate Professor in the Department of Internal Medicine, to introduce us to the first speaker. Ma'am, please.
Thank you, Siddharth. Thank you, Siddharth, for that introduction. Am I audible?
Yes, ma'am, you're audible and visible.
Yeah. Am I visible? Okay. Yeah, I'm privileged to introduce Dr. B.R. Yamuna Devi. She's an associate consultant and infection control professional, operation theater and central sterile supply department coordinator at Apollo Main Hospital, Greens Road, Chennai. Ma'am has wonderful educational qualification from MBBS from Maharashtra University, and followed by diploma in anesthesiology from Madras Medical College, followed by MD microbiology in April 2015. She has bagged the gold medal in her MD microbiology exam conducted in 2015 from Tamil Nadu MGR University. Madam is a prestigious recipient of Asia Hospital Management Award in Clinical Service Project in 2017. She has also emerged as a winner of Asia Pacific Society of Infection Control, CSSD COE 2018 award in gold category. She is also selected as an international ambassador for the Society for Healthcare Epidemiology of America in 2021. Her research includes plenty of ICMR funded study, also Center for Disease Control and Prevention funded national level study on hospital acquired infections. Madam is an author of multiple national and international journals and books. She has authored Prevention of Healthcare Associated Infection and API textbook of medicine for medical students. She has also published a clinical key essentials Elsevier publishers on point of care solution for CLABSI, central line associated bloodstream infection. Madam has presented an umpteen number of journals. Madam has also presented as speakers at multiple CMEs, at multiple conferences, at national, state level, international levels. Sorry, I couldn't play the slide that time. This is ma'am's guest lectures at multiple places, even in 2020. The year of pandemic, Madam continued to show her professional skills. Her research publications are ultimate. She has multiple publications. Thank you, ma'am. Over to you. I'm pleased to inform and pleased to introduce you. A request to the delegates, kindly put in the questions in the chat box so that we can answer at the end of the session. Ma'am, you may start now.
Thank you. Thank you so much for the introduction and for the opportunity to be a speaker for this outbreak management session. I would like to share the screen. Is my screen visible? Is it clear?
Yes, madam.
Yeah. So my topic for today's CME is approach to unknown outbreak and seeing beyond the horizon. Going with an example, in an 80-bedded pediatric hospital, a patient with a cardiac anomaly underwent corrective surgery, post-op AKI, hemodialysis, peritoneal dialysis, presented with fever and blood culture grew ralcytonia. No other case for the next 1 month and no other case since last 2 years. Now, what will you call this as? First case of an outbreak. No, it's an incidental case. Call your micro lab and check with them. Call your neighboring hospital and or start your not 8 work. Again, one more presentation is about in a dialysis unit, one of the patient or two of the patients got converted to HCV positive, previously who were seronegative. All other cases were negative, 5 machines were used, and 1 machine was used for HPV and HCV at the end. But there was no physical separation from the rest of the negative patients. So what will you call it as? A HCV outbreak, incidental finding? Should we do an RCA? Should we check all the employees? So the objective of today's talk is why we need to know what do you mean by an outbreak, steps involved in an outbreak and how to proceed it, and at the end, how to summarize. Outbreak is nothing but less than a CID investigation where the hidden agenda or the hidden your criminal is of course an invisible agent, which is a microorganism. It is not new to us. We all know about this investigation happened way back by John Snow, where we found that in the Broad Street, the pump A was the main factor contaminated with cholera, which led to the outbreak of cholera disease. Now, why to investigate an outbreak? We need to, of course, identify the source, eliminate it, look into the severeness of illness, which has put all the patients into critical stage, develop important strategies to prevent further outbreak, and evaluate the existing and implement preventive strategies, describe new diseases, research insight, and learn more about new diseases, like what we have done in the beginning of COVID-19, of course, even regarding Nipah virus, which was, of course, an outbreak for all of us in the beginning. Address the public concern, and of course, it's our job. Why outbreak investigation? Outbreaks leads to increased morbidity, mortality, prolongation of stay for the patient in hospital, additional procedures, quality of care gets affected, and increased cost of the patient, and you need to answer multiple people. So the basic principles became behind this outbreak investigation is you have to be systemic. Follow the same steps during every type of outbreak. Write down the case definition. Ask same questions to everybody. Stop often to reassess what you know. Line list your findings. If possible, put an epidemic curve and always coordinate with partners. Partners, I mean, is you need to know the site of crime, and you have to be behind the site of crime and involve the people who were there at the site of crime. So it could be the surgeons, the anesthetists, it could be the general physicians, it could be anybody at that place, the nurses, environmentalists, epidemiologists. Involve as many as people possible to crack the outbreak. One most important thing to remember is outbreak investigations are neither linear nor orderly. Multiple steps happen all at a time simultaneously, and the patients start presenting to you. And steps often have to be repeated. So the process of outbreak steps from 1 to 10 has to be repeated several times to achieve the findings. And before you begin, always talk to the lab. Tell them to save all the isolates that might be the cause of the outbreak, so that at the end you can do a molecular analysis. The principle of outbreak depends on incubation period, mode of transmission, temporal, geographical clustering, and of course the chain of transmission. If we put COVID exactly into the place, what they had to understand from the beginning, the scientist, is what is the incubation period. That helped us to define the period of quarantining. Mode of transmission, is it a droplet, airborne, contact, indirect, waterborne? Yes, it helps us to break the chain of infection. Is it clustered in some part of the region, or is it clustered in one part of the zone? Yes, you have to contain that zone. And what is the chain route of transmission? So the steps of investigation goes first to form a investigation team, where I already told involve multiple partners, generate resources which is required. It could be lab related resource, it could be manpower, it could be technical resource. Contain all your resources. Confirm the existence of an outbreak, where in the subsequent slides we will see what is an outbreak, define an outbreak. Verify the diagnosis, whether it is really an outbreak or not. Define case and conduct case study. Find the cases systematically, put them in line list, tabulate, and take immediate control measures. Never wait for the outbreak that you will investigate completely, then you will start the control measures. It should be in between, and it should be immediate. Formulate hypothesis about how causes of illness and source of infection has happened. Evaluate the hypothesis with analytical methods if possible. Plan and execute additional studies, like it could be a surveillance study, laboratory based study. Develop and implement preventive and control measures, and communicate. The most important thing, communicate to the people on the floor and maintain the surveillance. How do you find an outbreak? Of course, by surveillance cultures, if you are following it up on a regular basis, you can, but you don't follow every surveillance aspect. So many things are picked up by a very good observant healthcare worker who come back and tell you that, yes, probably I see there is an outbreak happening in the place. So the definition of outbreak is occurrence of more cases of disease or condition than expected in a given area among a specified group of people over a particular period of time. Two or more linked cases, or increase in clinically relevant with important public implications. So when you see even one case of smallpox or even one case of polio happening, it is of course an outbreak. So number doesn't matter. Even one case, if it has never happened in that place, in that specific area, and within that specific group of population, it would be called as an outbreak. The common detected outbreak in a hospital is nosocomial outbreak, like related to MRSA or a carbapenem resistant organism, Enterobacteriaceae, Pseudomonas, Clostridium difficile. At times related with the kitchen, like Salmonella outbreak, Campylobacter, norovirus, respiratory pathogens, influenza, measles, chickenpox, or very, very rarely Legionnaires disease. So this is example of a pseudo outbreak of cryptococcal meningitis, where they found that patients for whomsoever CSF sample was sent always had a cryptococcus presence in the CSF sample. The reason, when they went back and saw whether it was really affecting the patient clinically, no, but only the CSF sample had cryptococcus found. The reason why, because to the CSF sample, they were adding a little bit of albumin, and this albumin, which was taken with the help of a syringe and a needle. In that needle, it was already stuck with some cryptococcus from the previous specimen, which got contaminated. And in subsequent slide preparation, it was contaminating the CSF. So this is what we call it as an pseudo outbreak, where there is no clinical evidence of patient getting infected. The second example of clinical pseudo outbreak is positive Klebsiella oxytoca cultures from the peritoneal fluid of 3 different patients who underwent paracentesis, and molecular typing was also done, which showed all the isolates are genetically related. We always think that if it's molecularly linked with each other, yes, definitely it's an outbreak. Yes, definitely these isolates are linked with each other. But is this an outbreak? No. The record reviews indicated that clinical presentation was not consistent with subacute bacterial peritonitis in any one of these cases. This was a case of pseudo outbreak. Again, for the blood cultures, it was actually supplemented with a medium of yeast extract and dextrose from a multi-dose vial. Now, this multi-dose vial was contaminated with Klebsiella, and subsequent fluids which was being cultured grew the same organism, which was not only phenotypically but molecular based similar to each other. So pseudo outbreak is nothing but resin positive culture without a clinically evident disease. When there is an artifactual increase in disease incidence without an increase in clinical disease, you always have to think, is it a lab related contaminant? Or probably you have an enhanced surveillance system which is picking up, or that could be an improvement in laboratory diagnostic method. Probably they used to have a manual diagnostic method, now it is an automated system, they are able to pick up more organisms. So that also could be the reason why you start seeing similar organisms multiple times. The first and foremost thing, before you start an outbreak, do a literature review. It is a very important thing to start from where this outbreak has started. There are lots of published articles about the outbreak investigation, and this is a very great resource from CDC, which has got all the outbreak database right from different countries, where more than 50,000 publications are available. So they have already did the hard work. What we need to see is, if you are finding an outbreak related to microorganism or related to any specific area, you just have to type it, find it, you will get n number of articles from this resource. So how do you find or verify the diagnosis? Usually it is from the microbiology data. Obtain the patient's clinical record. You can get also the culture record or the surveillance record from the infection control team. Rule out misdiagnosis or any lab error if it is happening. And then, of course, discuss with the clinicians. Do they see any kind of clinical evidence or disease among the patient? So what are the different case finding challenges, even if we have a clinical record of the patient? Finding cases when you can't rely only on microbiology, and it is very tough, and it requires even a lot more effort to chart the review. It requires always a link between case finding. Yes, microbiology lab always supports you to make the case finding easier, because it is always easy to go retrospectively with the help of the organism rather than to looking prospectively into all the medical records. But you may miss cases. For example, all it depends on the pathogen. There can be subsequent difference between a influenza positive versus influenza like illness. Diagnosis do differ, but organisms can be similar. In some instances, there may be cases with subclinical infections, or they may be colonized also, but that could be the reason of transmission of infection, because colonized patient doesn't mean they don't transmit infection. Will surveillance culture help us to know cases, and is it the worth extra time and money? Remember, always the goal of investigation is to stop the outbreak, and it is not to uncover every case which comes on your way. It is more exhaustive case finding efforts may not be needed upfront, but might become important if you are not able to come to the core reason why an outbreak is happening. The fourth point is, how would you define a case and conduct case finding? It should have 3 important components: person, place, and time. For example, if there is a staphylococcal food poisoning outbreak, define a person means it defines an acute illness involving fever, vomiting, and diarrhea. Place, it is happening in near or people residing near the restaurant A. time during a period of June 2016, after a period of 6 to 8 hours when they came in contact with restaurant A. So this completely puts together all the things as a case definition where you can share with a clinician, other people in the department. So if at all they find such a case, they will be able to report it back to you saying that, yes, this is an outbreak. So the purpose of case definition is to put all three things together: person, place, time, evaluate the information, and then look for the pathogen, source, transmission, and postulate your own hypothesis from where this could have happened. It's important about line listing. So line listing, what we mean is it depends on the different types of case. It could be like signs and symptoms of patient, since when, type of symptoms, onset of date and time, what were the medications the patient was on, the different procedures, consults, location, staff contact, and host factors. Gathering line list information is the most intensive part of the investigation team, but if you do it, you will definitely be able to crack the outbreak. Even a limited line list sometimes will help you. For example, a Pseudomonas outbreak where all cases underwent bronchoscopy prior to infection. You may not know that multiple of your patients on ventilator will be growing Pseudomonas, but when you start culturing things, you find nothing is growing Pseudomonas. But when you see a common factor that all these patients underwent a bronchoscopy brought from the bronchoscopy unit, probably it was not cleaned properly, will clinch you a diagnosis for the outbreak. Sometimes the limited line listing can also be misleading, and not every case might be exposed to the source. For example, if you take the line list of patients that all patients were on ventilator, but you forget to see what were the doses of drugs where the patient was on. Probably the albuterol which the patient received was the source of infection for the patient, where you have to see it more meticulously. So everything is there. The devil is always there in the chart. We had this outbreak scenario which happened that Burkholderia capacia was growing in the patient, but after extensive work, we couldn't find it out. But when we started having a group discussion with the CCU consultants, we came to know that they are using a new product of video laryngoscope, which they were not sterilizing it because it was a new product. It was a company given product. They do not want to break it. So they were just using it for the patient, washing it under tap water and storing it by the side. So that was a new intervention which happened in the hospital. So always discuss with the people working on the ground. Are they trying out something new which you are not aware when you are doing a line listing? Another outbreak we had was the dialysis patient who were undergoing dialysis grew Burkholderia and Ralsitania picketi. So we were looking into the dialysis water. It grew nothing for us. Then we looked into the endotoxin level. For that area, yes, the endotoxin level at the port of the patients was a little bit elevated. So we were like not able to find the reason why. Is it a cross transmission because of hand hygiene? The dialysis port was from a cheaper version. So the engineering team changed it long back, which none of us were aware about it. So when we removed that valve and cultured, it grew Burkholderia, Cenocepaceae, and also Ralsitania picketi. So that was the main reason for all the patients who were staged in that particular wing grew this bug in their blood. So epidemic curve or analytical method analysis helps us to know from where the source of infection has started. Is it a point source or is it a propagated infection? How is the peak? So when was the maximum number of cases happened? So where are we going towards? Is the curve coming down? Is it a sharp curve? So all the analytical method helps us to find the exact incubation period and we are in which phase of the outbreak. So describe start and duration, peak, importance, atypical cases, and it helps to develop a hypothesis. So this is a second epidemic curve B, which tells us about that it is a propagated level of infection, that it moves from one person to other, and it is still ongoing outbreak and you have not curtailed it. So do you need to do one? In many cases, analytical study is always icing on the cake, but not necessary to control the outbreak. If you want to publish something, yes, it is always good, but not to control outbreak. They can help in guiding you more investigation when the resource remains unclear for a while, and they help you to support the hypothesis when there is no smoking gun. What is the things you should consider is, do I need to restrict potential controls to patients with longer stay? If you have to think about analytical study, you have to divide into case and control. So who should be the potential control? The patients who are staying in that area for longer duration of time. Do I need to match for a particular hospital location, only ward A or the wards near ward A? Do I need to only include controls with cultures obtained that did not grow the pathogen I'm investigating? So these are the group of controls which you can compare with the existing case of outbreaks. Aside from the patient, there will other interested parties. If you are working in hospital and if it is a big outbreak like dialysis patient ending up with HCV positive, it is a nightmare for the nephrologist who are working there. Not only the relatives of the patient, the management will be behind, the hospital administration, media people will be behind you, and medical legal lawyers will be behind you. Why this patient has seroconverted when he is coming to your hospital for several years to get dialyzed? So that means the sources from your hospital. So you have to be very crisp and fast in your outbreak analysis because you need to answer multiple people. So what is a good, bad, ugly for an environmental culture, which is the next step? It can be the most powerful and definitive aspect of a investigation, but it is more expensive, misleading, and sometimes frustrating because negative culture doesn't mean that the bug was never there. Or else, did we get the right culture? Did we do a right process? So methodologies are always tricky and might not be the standard method used in lab. So always discuss with your lab if you are planning to do a culture from particular place, what is the special media required, discuss with them, ask them to prepare the media, keep ready, or call them over to that particular place to do the culturing. So some samples result in overgrowth of the pathogens which you are not looking for. And some samples requires a neutralization step to get rid of the disinfectants in that place. Always culture you have data from the line list and observation. Whenever there is an outbreak, don't call the microbiology lab or you don't take the culture plate and start culturing all the places. No, first line list, identify what could be the source, what could be the mode of transmission, then you go for culturing. Talk with the lab about the optimal methods, store all the isolates of outbreak, culture only things that are likely your routes of transmission. Don't go and start swabbing every part of wall and floor of the hospital. Culture makes sense for the organism. If it's ralsitania or Burkholderia, look into fluids, water used on the patient. If it is VRE, always look for high touch surfaces, objects and surfaces. Remember, the environment is a very big area and your swab is very small, so you will end up finding nothing. Ultimately, the primary goal is to stop the transmission and not necessarily find the goal. It's okay to implement variety of control measures on the way and targeting various possibilities based on the initial observation. Implement and evaluate control measures. That is the important step. Prevent further exposure. Work with regulators and health educators to institute it. Communicate your findings. At the end, summarize your investigation, make appropriate recommendations, disseminate the report to all informed concerned person, prepare your preventive steps in the hospital and results of investigations. If it is something, a great outbreak happening, always transfer this information as a public information to the media also. So the idea of sequence of events is to curtail the yellow curve. What we did even for COVID is to broaden the curve, to lessen the curve. And the main step of outbreak is make the curve to the redder side and potential cases can be prevented. The yellow aspect of the bigger curve will be prevented by proper response to the outbreak investigation. Maintain your surveillance, deciding if outbreak is over or not, and documenting effectiveness of control measures. Always keep finding it again and again. It doesn't mean the outbreak has ended. So you have to keep a follow-up, whether it is recurring again. Is your control measures adequate enough to prevent or control the outbreak? So this is one of our outbreak which we did in the year 2016 and 17, where 5.5% of our patient had bacteremia per 1000 CCU days. We had extensive line listing of all the patients. We looked into all the common sharing item between the patient, right from ultrasound machine, the ECG used on the patient, the cardiac monitors, the bronchoscope, are they ventilated patient. And then ultimately we found the gel which was used on the patient, is it a common source of infection? Because gel doesn't penetrate the skin of the patients, but still we were like, why not? This could be a source of infection. So when we cultured the ultrasound probe gel, it was so contaminated that it grew Burkholderia capaceae. Probably these Burkholderia capaceae are very difficult enough. One, it sticks to your skin. So that point of time, if you're going to do any invasive procedure via the skin, that called bacteremia for the patient. So this was our outbreak curve. So once the gel usage was removed from the hospital, we were like able to curtail completely Burkholderia capaceae bacteremia outbreak in the CCU. So ultimately the summary: detection, effective surveillance system, vigilant hospital personnel. You should have a good rapport with them who would come back and tell you, I smell an outbreak there. Ask the lab to save all positive culture, have skilled hospital infection control practitioners and microbiologists and epidemiologists, prevention, appropriate infection control practices, and strategies to prevent and control antimicrobial resistant pathogen, because that is the commonest nosocomial outbreaks which happens in the organism. So the ultimate goal of every individual or every clinician is only the patient safety. Thank you.
Thank you, ma'am, for your brainstorming session on detailing outbreak investigation in simple terms. We have a couple of questions. Can we take one by one?
Yeah, please.
The first question is, are outbreaks likely to repeat if no environmental changes are done for it?
Yeah, definitely. So what I told is, for outbreak to be curtailed properly, the source of infection has to be found. If your source of infection, you are not able to find it, then of course your outbreak will not be a point source, it will be a propagated. For example, as I mentioned, the ultrasound gel, the simple example, probably for a patient in bed number 2003 and 2004, the ultrasound gel would have been used. But for patient in bed number 5 and 6, it would not have been used. But from this patient who is colonized with Burkholderia capaceae, if you have a poor hand hygiene practices, it gets transmitted from one patient to other. So what is more important? To find the source of infection, then only the outbreak will be curtailed.
Okay. What are the likely characters of an outbreak which indicates chances of pandemicity? That's the second question, ma'am.
What are the
likely characters of an outbreak that indicate pandemicity?
Okay, so characters of an outbreak, if I say, is whichever is an airborne, the mode of transmission. So for pandemic, things has to happen. It always depends on the mode of transmission and the susceptibility of the host. If your mode of transmission is of course air, then the pandemicity becomes very common.
Ma'am, how do we say that something will become endemic?
How do we say when something becomes endemic?
Yes, ma'am. How do some diseases become endemic?
Yeah, for example, we can't say communicable diseases has endemic in a hospital scenario. It's a communicable disease. But certain nosocomial infections, when you see like Acinetobacter carbapenem resistant, so you do receive patients from outside hospital also comes with colonized carbapenem resistant Acinetobacter. So that point of time you call it as or carbapenem resistant enterobacteriaceae when you receive patients from outside. So when the numbers within the hospital, if you have 20 beds, and in that 20 beds, 18 of your patients already are carbapenem resistant, you call it as endemic. But endemicity term as such for a nosocomial, you can't put it. So probably you can say that you missed 1 or 2 patients. That's the reason why it has become 18 patients now. But endemicity in a nosocomial outbreak is very difficult to define. In a communicable disease, in public health point of view, when it is common, for example, we don't call malaria happening in our country as a outbreak or an epidemic. No, it happens during that seasonal time. We call it as an endemic that point of time. So it all depends on a serial of infections over the past 5 years when you see, and in that particular season, if it is within the same range of numbers, then you call it as an endemic. If it is beyond that number, then you call it as an epidemic even for that season.
Ma'am, the next most anticipated question, what in your belief will happen to corona epidemic in India? Do we have any hope?
Sorry, I'm not an expert in that field to define what will happen to corona. I'm not an epidemiologist, sorry.
Okay. Ma'am, my next question would be, how much of case detection is required to characterize a disease as an outbreak?
Yeah, as I already mentioned, it is like you have to start from somewhere. So the easiest place to start is the microbiology lab. So if there is a Serratia bacteremia outbreak, which has been happening in the month of July, so you get all the Serratia bacteremia which the lab has reported, line list it all together and see in which ward it is happening. If it is in your surgical HDU, line list that entire surgical HDU, look into that entire Serratia bacteremia. So it is always either a syndrome-based outbreak. If it is diarrhea in a patient happening in, for example, surgical HDU, you look into the all diarrheal patient. It all depends. Either it is a syndromic approach you are going to do, or is it a bacterial or a microbiological related outbreak. So if it is a syndromic outbreak, go from the medical records of all the patients who were admitted in that particular area for that period of time. If it is a microbiological outbreak, get all the details and then go retrospectively with your clinical records.
Ma'am, one more question. If we notice a cluster of cases and we are not able to identify any source pathogen, that means host environmental factor for the same, whom should we approach to investigate? Is it any local body whom we can approach, or any person for a state or somebody nominated of that sort?
Yeah. As such, our country doesn't have those kinds of bodies where we have. Of course, in US and all, they do have CDC, so they can call the CDC and tell them if it is an ongoing outbreak causing multiple healthcare associated infection, causing public health importance, you can call the CDC. Whereas as far as India goes, we don't have. Probably we should involve the state board over there, the people working in the corporation of your particular area. You can involve them and ask them to investigate. But again, it becomes a big issue for us. So because if you are not able to find it and you involve external people, they will start digging you in a multiple way. So as far as possible, you try to crack your own outbreak. If not, you can always include the healthcare authorities from the government aspect to come and help you out.
Okay. Thank you so much, ma'am. We were very happy and very glad to have you here, and a very lively interactive session we had with you. Thank you, ma'am. Over to you, Siddharth Pandey.
Thank you once again, Dr. Yamuna Devi, and for the amazing presentation, and Dr. Supriya P.S. for moderating the session. Thank you once again.
Thank you.
There is nothing so patient in this world or any other as a virus searching for a host. I now invite Dr. Pratiksha Rai, Assistant Professor, Department of Internal Medicine, SHEMA, to give us all a brief introduction of our next speaker, Dr. V. Ravi, who will deliver a lecture titled Vandalizing Viruses. Ma'am, please.
Yes. Good morning, everyone. Myself, Dr. Pratiksha Rai, Assistant Professor in the Department of General Medicine, SHEMA. When the pandemic hit in March 2020, one person Karnataka depended on to stem the tide was Dr. Vasantapuram Ravi. Virus Ravi, as he is fondly known to many. Dr. Ravi's career has taken him through 35 years of extensive research and clinical experience in diagnosis, treatment, and public health aspects of viro-neurological infections. He pursued his MD microbiology from JIPMER Pondicherry in 1983. Dr. Ravi established Department of Neurovirology at NIMANS on 28 January 1985, which was formally inaugurated by the Nobel laureate Dr. Baruch Samuel Blueberg. Just to mention few of his numerous contributions, he is one among the first to develop indigenous ELISA kit for specific diagnosis of Japanese encephalitis virus, neurocysticercosis, HIV. Dr. Ravi was tasked with crucial responsibility by ICMR for mentoring all private and government laboratories across the state to establish COVID-19 RT-PCR testing facility. As expected, he was successful in having more than 160 laboratories approved in less than 3 months which helped to ramp up testing to more than 1 lakh per day in state. Dr. Ravi's team has been among first few in country to sequence entire genome of SARS-CoV-2. At NIMANS, he served as a senior professor and head, registrar, dean of basic sciences, control of examination, and retired in November 2020. He's also an NABL assessor since 2012. He has been principal investigator for several funded research projects. He has published in more than 160 journals, contributed 20 chapters to books and 4 WHO monographs. He has mentored more than 25 PhD students, several MD students, and junior colleagues. Since 1990, collaborated with the University of Liverpool, also served as a co-director of Dual Lemons Liverpool PhD program. Shining feather in his cap is being elected as a distinguished international fellow of American Society of Tropical Medicine and Hygiene, National Academy of Sciences, Indian Academy of Sciences. He is nodal officer for genome sequencing of SARS-CoV-2 in Karnataka. Also member of Karnataka COVID Technical Advisory Committee, COVID-19 Vaccine Committee, ICMR Expert Group on COVID-19. He was recipient of several awards. To name a few, C.V. Raman Young Scientist Award from Government of Karnataka in 2000, ICMR J.B. Srivatsa Oration Award, etc. We are in for a real treat this morning. Please join me in giving me a warm welcome to Dr. Ravi, who will be talking to us on the topic Vandalizing Viruses. It is an honor and a privilege for us, sir, to have you with us today. I'm sure we will be enriched by your talk. Without further ado, then over to you, Dr. Ravi.
Good morning, and let me at the outset thank the organizers of Nitte University and SHEMA Academy, Medicine Department in particular, for inviting me. This is my third online visit to Nitte. Of course, I've been there a couple of times, and I have very good friends, both Dr. Karuna Sagar and Dr. Indrani Karuna Sagar, whom I have known for many years. Now, what I will do, I've been asked to talk on viruses with an outbreak potential, with a very interesting title known as Vandalizing Viruses. I hope you're able to see my screen. Can somebody confirm?
Yes, sir, we can.
Thank you. Thank you. What I will do in the next 45 minutes is to talk to you about, I'll start from the basics because it is a CME. What are viruses? Why are they here to stay forever? Where do they exist? And more importantly, how we should learn to live with them, because we're not going to get rid of viruses in this universe. What is the role of laboratory in outbreak investigation? What are the viruses that have outbreak potential in India? What do we know so far? What is the history? And then end with a summary. Now, viruses by definition are ultra microscopic. You can't see them in ordinary microscope. They have a single strand of, they have a single nucleic acid, not single strand, either RNA or DNA. And they're surrounded by a protein coat. One of the most simple organisms structurally in the universe. But the difficulty is they're absolute parasites. They can multiply only within living cells. So there is this parallel perpetual argument: are viruses the most evolved form of life or the simplest form of life? And I think that will keep continuing the argument. Sir Peter Medawer actually summarized virus in a definition in a single line by saying it's a piece of bad news wrapped in a protein coat. Why are they here to stay forever? God endowed viruses with several inherent advantages because of their simplicity in structure. They replicate very rapidly after entering a cell. In fact, the half-life or the multiplication of viruses outbeats all other living forms. So millions of particles produced in an hour. RNA viruses are endowed with the property of mutation. That's because the enzyme that copies the genetic material of RNA viruses, RNA polymerase to be more specific, makes mistakes while copying. In simple terms, it lacks proofreading ability. Now you're seeing a very classical example in front of you in the SARS-CoV-2, how it is continuously, it surprised everybody. Nobody knew coronaviruses will mutate so fast. Influenza is a classic example of mutation, and so is HIV and so many other RNA viruses. While DNA viruses are endowed with the property of latency. Now latency means lying dormant. And this happens because they have the ability to integrate into our genome, host genome, be it animal or human, and there it lies dormant and activate itself when the conditions are favorable. And lastly, COVID has brought to focus as to how viruses jump species. We all know now that viruses from animals and birds and other mammals can jump to human beings. Now, this is something called a spillover. And a spillover occurs because of the selfish nature of human behavior. I would attract your attention to a very important book that's published. It's available in Flipkart or Amazon. And those of you who are interested in reading should read this book called Dancing Matrix, which traces beautifully the evolution of how viruses emerge and cause outbreaks. Viruses live in their own corridors is what K.S.HEGDE, the author of this book, argues and says that it's man who repeatedly crisscrosses these corridors and invites outbreaks. They are abundant in nature. I don't know how many of you know, 60% of the ocean mass, if you have to wait, is viruses. They're very, very important for biodiversity and for evolution. And they are present on all the flora and fauna of the planet, be it plants or animals or birds, or what you have. Whenever human beings are in very close proximity to places where they should not be, or to animals they should not be, then that's when the spillover occurs. So there is interesting, there's a lot of interest in the last few years, and more so now, in the concept of One Health. One Health is taking care of the environment, the animals and birds, so that human beings can be healthy. Unless we respect the environment and the other living forms, if we don't, then we will have pandemics like COVID. So spillover of events continue to occur in many parts of the world. The viruses jump from rodents, bats, and non-human primates to human beings. And this is because of proximity and crowding. I'm showing you pictures of some of the wet markets in Southeast Asia. One is in Vietnam and the other is in Wuhan. And we all know that bats are the reservoirs and rodents are the reservoirs for many viruses. In fact, for SARS-CoV-2, all the severe acute respiratory syndrome viruses like SARS-CoV-1, MERS, and SARS-CoV-2, they all, the reservoirs are bats and they spilt over through intermediate species. In fact, if you look at the past decade, it has been very clearly established that most of the outbreaks that occurred in various parts of the world are because of spillover from bats. And more bat species are being described, and we know that bats are one of the major sources of outbreak viruses. They can infect humans, they can infect birds, and they can infect animals. Therefore, we have to learn to live with viruses. There's no way we are going to get rid of viruses. And I'll put the argument differently for you, saying that an estimated 10 nonillion. Now, what is a nonillion? 10 to the power 31 is a nonillion. We all know up to trillion, billion, and trillion, not more than that. But this is a nonillion. That is the estimated number of viruses that exist in our planet. And enough to assign one to every star in the universe 100 million times. Mammals and birds alone are thought to have 1.7 million undiscovered types of viruses. Now, so they are widely present on living forms in the earth. More than 200 of these viruses are known to cause disease in humans, and many of them, more than 24 of them, are capable of causing large outbreaks. Of the pathogens that have infected human population in recent decades, you will see that outbreak potential is almost the exclusive property of RNA viruses. You hardly have ever seen a DNA virus like herpes causing huge outbreaks with epidemic or pandemic potential. Yeah, chickenpox is DNA virus. It can cause outbreaks within hostels and educational institutions and schools. But they are very focal, localized outbreaks. Large scale outbreaks sweeping across the globe is more associated with RNA viruses. More interestingly, 10% of the human genome is made up of bits of viral DNA, and these are referred to as endogenous retroviruses. So viruses are here to stay. Now let's learn to respect them and learn to live with them. If you look globally at the kind of viral outbreaks, epidemics, and pandemics that have occurred, now this is a picture I took from Nature reviews in immunology several years ago. Then, of course, I've modified to add the other viruses that have come on after 2013. These are the tombstones, meaning the number of people who died because of viral outbreaks. Starting from the Spanish flu, of which we have a great record, then to West Nile, to Ebola, to HIV, which is still ongoing, and then the Nipah and the SARS, and the MERS and the Ebola and the Zika, and of course the latest being COVID. So over the past century, humanity has witnessed an emergence of a large number of viruses. I joined the medical school in 1973, and I had the privilege of seeing so many new viruses being discovered, and many of them are actually caused large outbreaks. In the last 48 years that have been in the field of medicine, 24 viruses. That means every 2 years a new virus is being ascribed to an emerging infection, starting from hepatitis B to SARS-CoV-2. Why do viruses emerge? What are the reasons for it? I mean, there are a wide variety of reasons. I told you some of them, that human behavior is the most important. There are political reasons, economic reasons, and social reasons why viruses emerge. Now, if you take the example of SARS-CoV-2, you will see that it was a spillover because of excessive crowding. And spillover events will continue to occur. And believe me, there are hotspots for spillover in the world. And many of these hotspots are in Southeast Asia, in the markets of China, Vietnam, Cambodia, India. India can be a hotspot. In India, it can be either the Northeast because of its biodiversity, its wildlife, and its terrain and human-animal contact being more, or in the Western Ghats. It could be African jungles, like Ebola emerged from there, or it could be the South American jungles where Zika and other diseases have emerged. So human behavior is the main reason for emergence of virus. There are, of course, genetic and biologic factors. The virus undergoes mutation or recombination. For example, the H1N1 outbreak that occurred was a result of recombination between bird, animal, and pig viruses. I mean, bird, human, and pig viruses. So, changing in agricultural practices, environmental factors. Building a dam, we had Congo hemorrhagic fever. Changing agricultural practices in Kerala brought Japanese encephalitis. And ecologic factors like climate change. Everybody is talking about global warming and expanding of the dengue borders. What was tropical is now subtropical. The borders of dengue are expanding because mosquito breeding is occurring in places hitherto that were cold have now become warm. Deforestation, KFD. A lot of societal events, war, conflict, the Ebola outbreak that occurred is as a result of people moving out of Rwanda in 1994-95. Healthcare devices can cause outbreaks. Food production, we have seen that in the greed to make the cow or the cattle more fat, they fed sheep remnants to the cattle. Cattle are vegetarian by nature. You feed them with offal, scrapey infected sheep material, and you produce mad cow disease. Sexual behavior, HIV, we know, and of course, a lot of other reasons like microbial adaptation, mutation in the virus in 2005, resulting in chikungunya sweeping across the globe. So there are many, many reasons. We will not go into it. How do we tackle viral threats? I think this is very important. There are 4 pillars on which we should rest our efforts to tackle viral threats. First is surveillance. We should continue to have robust surveillance. What happens is whenever there is an outbreak, people get interested, and in between outbreak periods, nobody is monitoring what is happening. So you need good surveillance systems, and surveillance systems, to make it economical, should be more syndrome based. Respiratory syndromes, jaundice, encephalitis, gastroenteritis, and febrile illnesses with rash. If we have set up surveillance systems, routine surveillance systems, then when an outbreak comes, quickly these surveillance platforms can rule out existing ones or pick up an existing pathogen or look for a new pathogen. We also need efficient diagnostics. Right now, till recently, we were in the era of monodiagnostics. Now we are having multiplex diagnostics, which helps syndromes. I'll give you an example of it in my talk, like the encephalitis chip that we developed. Building laboratory networks. No one lab can solve the problems. So you need a network within states, within countries, that can quickly test and say this is a new pathogen or it's an old one. And of course, we need to understand disease biology, how it occurred, what it causes, what are the symptoms, so that we can definitely immediately provide relief to patients who are suffering from outbreak. The laboratory is of paramount importance during an outbreak, and it plays a great deal during outbreak in case detection. Most importantly, as Dr. Yamuna Devi was mentioning, specimen collection, then confirmation of the case, in case reporting to the systems, and support it can provide in terms of training to the field staff, and also provide guidelines and standards for. So in short, the laboratory has many, many roles, and I am listing some of the roles of the laboratory in outbreak investigation. As I said, detection and confirmation, guide the outbreak investigation by telling them what is the correct specimen to collect, when to collect, and how to transport, what details are required, do immunological studies to know if it's a population is naive or it's an existing pathogen, typing the pathogen, the genetic sequencing nowadays is very important to detect emerging variants, and feed in data to public health officials in order to change policy, which we did in 2009. We told the government that almost every patient we have tested were H1N1 positive. There's no pointing in a syndromic approach. There's no pointing in holding back oseltamivir. And that really helped curb the outbreak. And of course, in helping in treatment in terms of sensitivity, and also environmental and food analysis. So the lab plays a great role. Remember that laboratory confirmation is important in an outbreak in the early stages. As early as you can get laboratory confirmation, that will decide how effectively you control the outbreak. Not every case needs confirmation, but if 10 to 20% of the cases show a particular pathogen, then in public health, we assume that the entire outbreak is caused by the organism, provided they all meet the case definition. Now, normally what happens in an outbreak is that cases start increasing in the first 10 days, then there's a huge rise, and then it takes about a month or 2 months and the outbreak subsides. Outbreak subsides for a variety of reasons. We know the cause, we know how to prevent it, and people are becoming more careful, they change their behavior. So this entire red mountain, by the time we get the result normally in an outbreak, the outbreak is over its peak. I think this graph, the next graph was also shown by Dr. Yamuna Devi. What we should be targeting is that we get laboratory confirmation in the first 7 to 10 days of any outbreak, so that the huge mountain of cases can be avoided. Now that can happen if only there is systematic surveillance and there is a laboratory investigation continuously supporting this surveillance. Remember, the right specimen at the proper time, collected properly, stored and transported, is the key to outbreak investigation. And broadly speaking, we should collect the specimen as early as possible, and depending on the syndrome, you collect a specimen. For example, for jaundice, you will need blood. For meningitis, encephalitis, you will need blood and CSF. For respiratory infection, throat swabs and nasal swabs. For fever with rash, blood, and if possible, other pathogens like enteroviruses, it's good to have a throat swab. Gastroenteritis, you need a stool specimen. So I think the guiding principle for what to collect should be the syndrome and specimen relevant to that syndrome. Moving on to emerging viral infections in India, I've started with some things that are our own property, not reported anywhere in the world. And first on them is K.S.HEGDE. And being Karnataka, I started with K.S.HEGDE because it's the most relevant pathogen. You all know that it is a tick-borne flavivirus infection. The ticks that transmit the infection to human beings, they are the nymphs actually. They actually, their natural cycle is they are present in monkeys. In monkeys which are found in the Western Ghat area. And these ticks, when they fall to the ground, and when man wades into the forest barefoot, not fully clothed, in the dry leaves, these ticks are present and they bite, and the virus is transmitted to human beings. So natural cycle is maintained in monkeys, and man, it spill over. We had no business to go into the forest, but if we do, then we get. Dr. Arun Kumar did some excellent work in Manipal, and he showed that now the Casanova forest disease is no longer restricted to 1 or 2 districts of Karnataka. It is seen all along the Western Ghats and in 5 states, in Tamil Nadu, Kerala, Karnataka, Goa, and Maharashtra. So all along you see, along the Western Ghats, that these ticks are present, these monkeys are present, and it causes a febrile illness with severe prostration, headache is very prominent. It's a biphasic illness, and you can diagnose it with IgM, ELISA, and PCR. There is a vaccine available. It is not very effective. There are efforts to make a new vaccine. Influenza, which is the next, always a potential pandemic threat because it has 8 different strands of RNA. These are pieces of RNA. Unlike all other RNA viruses, it has 8 pieces of RNA. So there is a potential to recombine, like it happened in H1N1. When a cell gets infected with 2 viruses of different species, then there can be recombination. And incidentally, the pig is called the mixing vessel. Pig can get infected with human viruses and with bird viruses. And you all know that the surface proteins of influenza, the neuraminidase and the hemagglutinin, occur in various types, and recombinations between them gives a great potential for emergence of a new pathogen. So we have to be on the watch for influenza pandemic, be it human influenza or avian influenza or any other animal influenza can spill over to human beings. The next is Nipah. Nipah is again a disease that threatened to enter Karnataka. It was there in Kerala, and first reported in West Bengal in Siliguri district in 2002, and this was the epidemic curve. As you can see, this was seen in 2001 and 2002, and very sharp spike. It is a disease that is transmitted by bats, frugivorous bats, and in Bangladesh and in Siliguri, it was shown that these bats come to date palms because they love the date sap, and while feeding into the date palm, they can also infect this sap by either through their saliva or their urination. And of course, in Kerala, it was due to the bite of fruit-eating bat. It is presumed that it spread to human beings. And then it's a highly infectious virus, spreads through the respiratory route, and finally causes encephalitis. So the illness was initially confused in Malaysia to Japanese encephalitis because of its similar clinical picture. So Nipah is one threat that can occur, especially in areas where there are bats, and that happens wherever there are a large number of fruit orchards. And Kerala being a country part of the country that where a lot of fruit trees are there and fruit-eating bats are there, it caused Nipah outbreak. Hanta virus is a virus that is associated with fever and renal syndrome. It has a great potential because it is life cycle is it in rodents, and these rodents can get infected, and they can the rodent urine contaminated can cause infections in human beings in the form of post-monsoon outbreaks. Although we have not had great outbreaks, it has been reported from India, and you can also get through inhalation through aerosols. CMC has reported a large number of seropositivity in serum samples collected, and they have shown that the Hanta virus is present in India. I will not go into dengue, it's a subject by itself. Dengue is also a disease with a huge outbreak potential. Last 2 years, thanks to COVID, with people not moving outdoors and urbanization and construction coming to a halt, mosquito breeding, even though it is happening, people are not exposed to it. So one would see a rebound of dengue infections very shortly. Chikungunya again is a very important outbreak virus, and we know that the virus mutated in the envelope in 2005, and that caused sweeping outbreaks for several reasons in India. One of the main reasons in Kerala, it started this outbreak, And this was because of the collection of rubber sap in open coconut shells that Kerala was doing. Mosquitoes Aedes breeds in that, and then Kerala brought an intervention of covering these coconut shells with cellophane sheets and a rubber band, a simple intervention, and now breeding has been controlled. But this is a virus that keeps causing outbreaks. Wherever dengue is there, chikungunya will be there because the vectors are common. The next virus that has been described to cause outbreaks in whole of Southeast Asia and parts of India, especially in Kerala and some other states, is enterovirus 71, which causes the hand, foot, mouth disease. A very typical lesion you will see in children of blisters in the hand, on the tongue, and in the feet. This is a post-monsoon kind of outbreak that you see. Almost all outbreaks that are vector-borne or enterovirus are post-monsoon. So this causes fever, and it also causes neurological syndrome. But the clinical picture is very, very useful in making a diagnosis. Chandipura is our own virus, found only in India, found in central India. I was associated with the investigation of the outbreak in 2003, and this is a sandfly mediated viral infection. Very interestingly, it's a midsummer encephalitis. Children sleep outdoors because of the heat in the peak of summer. They are bitten by sandflies. It is seen in Telangana, it is seen in Maharashtra, western Maharashtra, southern Madhya Pradesh, western Gujarat, and in Chhattisgarh. That is the belt where it is found. Typically, very dramatic illness. Children are playing, they go to sleep, next day morning they wake up, they have fever, and within few hours slip into altered sensorium, high mortality, 50%. And the main pathology is massive cerebral edema. You can diagnose it using IgM capture ELISA kit, which NIV has made, and also by doing PCR on the CSF. Crimean Congo hemorrhagic fever has entered India. We know that more than 8-10 years ago, the NIV showed that this virus causes a hemorrhagic illness, first reported in Gujarat and also reported from Rajasthan. It's a tick-borne disease, and the cattle are the reservoirs in India, and this has a potential to become a big outbreak disease if not detected and controlled early. MERS, we are lucky that we did not have a MERS outbreak. It is a disease that was reported in the Middle East. Ebola, again, we are so far been lucky. Ebola's main hub is Africa. It keeps emerging in Africa from the African jungles. The highly contagious pathogen, close contact is known to transmit through body fluids, and it's a very, very dramatic illness, high mortality. Mortality can be as high as 90%. So this is again, so hemorrhagic fevers like CCA, Crimean Congo and Ebola, one has to keep an eye on. Apart from, of course, dengue and Zika, which also can cause hemorrhage. Zika has been reported in India. Fortunately, it has not caused serious outbreaks. It caused massive outbreaks in South America, and there was an epidemic of microcephaly as well. Now, Zika neutralizing antibodies were reported in India way back in 1952 by the Rockefeller Foundation, the erstwhile NIV. And Zika cases have been reported in India periodically. This year also we had a few Zika cases reported from Kerala. So it has been reported from Tamil Nadu, Gujarat, and Kerala so far, and Rajasthan, of course. So moving on, one of the main things that people are doing now, this is looking into the future, is to employ the power of IT. Now, Nathan Wolf is a guy who worked a lot in African jungles. He is a faculty at Stanford. Now, he set up what is known as listening posts. What are listening posts? He has chosen areas where virus outbreaks usually are reported. And then he uses IT information, big data analysis to find out, mining data from social media, from emails and other places, and trying to put data together and see if somebody says, I'm not coming to office today, I have a cold, then using geographic information systems, they try to predict flu trends. This is established during H1N1 and is continuing to do it. And then they home in and they find one particular area in a city, a large number of infections are, people are falling sick. Immediately they activate the public health surveillance system. They contain people. They've been able to avoid small or big outbreaks. So this is something good to identify hotspots. It's a different kind of approach using IT data and big data, social media data to predict outbreaks. We ourselves did a huge amount of work in Uttar Pradesh, West Bengal, and Assam, where we were asked to investigate the cause for encephalitis outbreaks that occur periodically. You know that until 2005, India had large outbreaks of encephalitis. It continues to have, but till 2005, majority of it was ascribed to Japanese encephalitis. With the introduction of the vaccination for Japanese encephalitis, JE contribution came down, but encephalitis cases did not decline. So we were invited by CDC to investigate this outbreak. We did a lot of things. I won't go into details. We engaged with stakeholders. We identified and built a network of labs in these 3 states. We finalized an algorithm. We built laboratory capacity in the districts as well as in the states for advanced detection, transported samples from districts to the city where the referral labs were there, and of course streamlined. So these were 18 districts where we worked in 3 major states where encephalitis outbreaks were a problem. And then we used an algorithm where the existing vector-borne disease algorithm was only testing for JE. To that, we added dengue, West Nile, chikungunya, and leptospira testing. And in the CSF, we started testing for bacterial meningitis pathogens by PCR, herpes enterovirus, and dengue virus by PCR. To cut a long story short, we tested 15,000 samples over 4-5 years in these 3 states, both by immunological assays and nucleic acid assays, using 6 immunological assays, namely JE, dengue, scrub, West Nile, lepto, chikungunya, malaria. We were able to get 45% etiology, whereas in nucleic acid test, by adding it only for CSF, we were able to get another 3.2%. Now, this total 15,000 patients. During the same period, these 3 states reported 37,000 cases. So 1/3 of these we were able to test, little more than 1/3. So this is a significant. And most interestingly, the commonest pathogens were JE, dengue, scrub. And in the later years, leptospira and chikungunya we identified also as the pathogens. So basically in this project, we were able to enhance, by using a simple algorithm, we were able to enhance detection by 3-fold, from 16% before we started the project to 48%. We also were able to put an algorithm which has now become the national algorithm for encephalitis testing. The ICMR and the vector-borne disease control program both use this algorithm, and also showed that scrub is a major pathogen in these 3 states. And once a patient is admitted and sample is collected, empirical treatment for scrub is given in all cases. Lastly, COVID-19 has taught us various lessons. And COVID-19, during outbreaks, it provides a great deal of opportunity to understand disease spread and disease biology. We did some simple analysis, which we published in Lancet eClinical Medicine last year. And this is looking at the efficiency of transmission between symptomatic and asymptomatic people. Now, what you see, this interesting diagram on the left, is a cluster analysis. A cluster is defined as 5 or more cases, and we identify the source, the index case that is in the center, and then from them how many people got it. As you can see, many of these clusters that you're seeing has a red dot. Some of them have a yellow dot in the center. Red means these were index cases were symptomatic. Basically, the point I'm trying to make is that symptomatic people spread infection to much more people than asymptomatic. And here is the hard data. Among the 46 clusters we studied, 32 were symptomatic, 14 were asymptomatic index cases. From 32 index cases, a total of 545 got COVID infection, whereas from 14 asymptomatic, only 114 got. So that's a stunning difference of 82 versus 17%. On an average, in the last column, you can see an asymptomatic person can give infection to 14 people, whereas an asymptomatic person can give it to 3 people. We also learned that COVID is not a direct viral disease. It is a disease that is mediated predominantly by the host defense mechanism. And the way the defense system responds, that determines whether you're going to be mild, moderate, or severe. So in summary then, human activities are the most potent driving factors for viral outbreaks and emergence. Classical examples are COVID, SARS, Ebola, K.S.HEGDE, I can go on and on. Social, economic, political, and climate change is very, very important to remember, and that is the one that contributes to viral outbreaks. We should harness technology like the one I showed you, the listening post, and the way we use technology during the COVID outbreak for tracing, tracking, quarantine app. All these are very, very important initiatives to contain outbreaks. And understanding and responding to disease emergence is very important. Having continuous surveillance in between outbreak periods is also very important. And in summary, the current global situation, unfortunately, the way we human beings are living favors disease emergence. Before I close, 4 books you should read if you are a bookworm, if you like reading books. One I already told you, Dancing Matrix. The other is Nathan Wolfe, where he describes this listening post called Viral Storm. Then Abraham Verghese, my classmate, who worked a lot on HIV, his book, which got the best Critics Award in America, called My Own Country, describing his experience with HIV. And of course, my all-time favorite, Paulo Coelho's novel known as Alchemist, which gives this very wonderful message that when you want something and it's for the good of everybody, entire universe conspires to give it. Thank you very much. If you have any questions, I would.
Yes, sir. Thank you. It was a very informative talk. We have few questions from audience. What will happen to our epidemic curve of COVID-19 in India? When we may see ray of hope? Can we ever be unmarked?
The road ahead for COVID is a road with multiple speed humps or speed breakers. The virus is not going to disappear as dramatically as it emerged on the globe. That is because until 80 to 90% of the population has either experienced infection or has received the vaccine, there will be outbreaks. And that is not going to happen very short. My own guesswork is that around this time next year, the virus would have become endemic. But you will see small outbreaks related to human behavior. Exactly what you're seeing in the last few days in Kerala. Following the Onam shopping, the crowding, and the festivities, there was a spurt. It was around 20,000. It went down to 10 to 12,000. It's bounced back to 30,000. I also believe that if we are not careful in our state, in the middle of September, second and third week of September, we will see a spurt. Post the festivities of all these festivals we are having now, exactly what happened in the first wave as well. And remember, the entire population is still not vaccinated. But vaccination is exceedingly useful. It reduces disease burden, severity, and death. But covering everybody with vaccine is going to take time. So I believe over the next year, we will see some more outbreaks, and then it will become endemic.
Can we anticipate the third wave of COVID-19 in India? Are children too susceptible to COVID-19?
My own personal opinion is we should anticipate a third wave. We should not make the mistake we made at the end of first wave. I believe there is scope for a huge spread. 50% of the state is immunized with one dose, close to 50%. That means the flip side is half of Karnataka's population is not immunized. Although seropositivity surveys taken with a pinch of salt say that 60% are covered, I would argue 40% is not covered. I am of the opinion that children are highly susceptible because they have been protected. Of course, their manifestation will be very mild. But given the number of children we have, I would say being prepared for an outbreak in children is very important. And also to use the opportunity to build infrastructure in hospitals for children. 60% of districts do not have a pediatric ICU. So I think expecting the worst is better than relaxing and saying it's already become endemic. We won't have a third wave. Pundits have told this at the end of first wave. Pundits have told it repeatedly in history. Look at history. Spanish flu had 3 waves. It would be foolish to assume there won't be another wave. If it doesn't come, well and good. But one should be prepared for a third wave, and that too in susceptible population, especially children. That's my take.
How much do you think India's infrastructure is prepared as of now?
India's infrastructure has phenomenally improved in the last 1 year, thanks to COVID. But for COVID, one of the best things that happened because of COVID is all governments and administrators, politicians woke up and started investing in health infrastructure. We deserved that long ago, but it is at least happening now. It has improved, and I hope we don't make the same mistakes at the end of first wave. We are ready with oxygen and essential supplies of drugs, and beds must be made available very quickly if there is a third wave.
Now, a lot of discussion about booster doses after 2 doses of COVID vaccine. What's your say on that?
There is absolutely no evidence to say you need a booster. There's a lot of hype, lot of concern, lot of anxiety. Studies are underway now. Remember, after 2 doses, immune system has memory, and it invokes memory whenever there's an infection. Antibodies are not the only measure of protection. Cell-mediated immunity also kicks in. So I would say it is too premature to conclude you need booster doses. I would say we'll have to have more robust studies. Longitudinal studies are going on. At the end of December, we'll have a very clear picture if we will need a booster. More than getting a booster to people, I think getting at least 1 dose to everybody is important because that reduces disease burden and also averts mortality.
Why vaccines are not protective against nasopharyngeal viruses? Do we have a nasal vaccine in the pipeline that can afford surface immunity, which might prove more effective?
There are many in the pipeline, at least one in India. Since I am a part of the DBT vaccine expert committee, I've been looking at the data. It's very promising, but it is not going to be available before the end of the year. Right now, these vaccines are very promising. How they will fare in phase 3 trials, we'll have to wait and see.
Why does COVID-19 produce extensive thrombosis and more often pulmonary thromboembolism? We have seen in our hospital who had taken 2 doses of vaccination, had presented to us with COVID positive status, developed a leg clot and thrombosis in spite of anticoagulation and had expired.
See, coagulation in COVID, as I mentioned, is the reaction of the immune system to the presence of the virus. And also remember, SARS-CoV-2 attaches and multiplies in endothelial cells, damages endothelial cells. When it damages endothelial cells, that's a nidus for clot formation. So this is something that happens usually in the second week of illness, and even post-COVID, it happens. So we have learned that COVID virus is capable of inducing thrombosis, not only through endothelial damage, but also through production of a number of cytokines that actually favor clotting. And this is also seen as a very rare side effect of certain vaccines, such as the viral vectored vaccines. And we have to live with that. But the benefit of vaccination far outweighs the risks of coagulation. So coagulation itself is a phenomenon that is mediated more because of the immune response.
What is your take on theory of man-made viruses?
I will not get into that controversy. It will remain a matter of great debate over tea and coffee, and I welcome that debate. At least it is brought out in the open that man can make viruses. I don't know if it's a man-made virus, but there is more and more evidence seem to suggest that it might have been a spillover from a lab. Whether it is engineered or not engineered, I think these are all lessons for us in future that we have to be very careful.
What factor would have helped in curtailing epidemicity of Nipah despite of its airborne spread?
Please come again. I didn't understand the question.
What factor would have helped in curtailing epidemicity of Nipah despite its airborne spread?
What factor can?
What factor would have reduced the epidemicity of Nipah despite its airborne spread?
You cannot prevent viruses from emerging. The only way is if you behave responsibly. Even if you behave responsibly, you cannot blame anything or anybody for Nipah coming to Kerala. But you should appreciate Kerala. They picked it up very fast and they contained the outbreak so beautifully because they were able to diagnose it and take appropriate care. A bat coming and biting you, you cannot prevent. But going into the forest, you can prevent. You need not get K.S.HEGDE. You need not get SARS-CoV-2. If you have respect for other life forms and if you still want to eat them, you want to sell them, do it in open markets. Closed markets, crowding, these are all things that will drive emergence of viruses and will be the root cause of outbreaks in future.
How can we differentiate microbial versus chemical induced outbreak?
I had the opportunity to work with the Muzaffarpur outbreak. I didn't bring it in here. Basically, one has to differentiate, at least in the context of CNS, I can tell you, encephalitis versus encephalopathy. In any corner of the world, if you use your clinical knowledge, you can do it very, very clearly. Itis is inflammation. That means there will be certain signs that are very typical of inflammation. And if you have access to lumbar puncture, it will tell you if there are cells in the CSF, then it is itis. When there is no cells in the CSF and it's a very dramatic illness and there are no focal neurological deficits, I would name it encephalopathy. And the Muzaffarpur outbreak, as you know, is a combination of a toxin from lichen and hypoglycemia. Both occur during a particular time of the year in the lychee capital of India at Muzaffarpur, where children, due to the extreme heat, play outside when their parents are harvesting lychees, and they consume hundreds of lychees, and they become tired and they sleep without a meal. And the toxin in lychee further compounds the hypoglycemia, and it produces seizures and mortalities around 20 to 30%. So apart from this, there is also another Saharanpur encephalitis which I was associated with. That is due to the consumption of a toxin from a plant, the seeds of a plant known as Cassia occidentalis. They are very attractive seeds that look like tur dal, and children eat it, and it causes, it's a very potent hepatotoxin. It produces hepatomyolysis and then results in. So the way you distinguish chemical from infectious would be clinical features, core laboratory diagnosis, as well as going to the field and studying the terrain, doing the line listing and looking at the environment around, the temperature, everything, and then concluding whether it is chemical or infectious.
So one last question. Will public education about prompt reporting of unusual symptoms help in containing outbreaks?
Absolutely, there's no doubt about it. That's something that is a dream. I don't know how much it will be a reality. Public participation is very important in outbreak prevention and outbreak control. And COVID has shown that it is very, very important, and public education is very important.
Thank you so much, sir. Over to you, Dr. Siddharth.
Thank you, sir, for patiently answering all our doubts and for the very lucid presentation. And my personal thank you for suggesting the books that you did. I'm sure some of the delegates will definitely go through the literature that you've suggested and gain from it. Thank you once again, sir. I now invite Dr. Shefali Prabhakar, Senior Resident in the Department of Internal Medicine, SHEMA, to introduce us to our next speaker, Dr. Nitin Gupta, who will deliver a lecture titled Mycosis, Lurking in the Shadows. Over to you, Dr. Shefali.
Thank you, Dr. Siddharth. Good afternoon, ladies and gentlemen. It's my great privilege and honor to introduce our next speaker. He's a talented physician scientist and also published author, Dr. Nitin Gupta. He has finished his MD Medicine, DM in infectious diseases. He has a diploma of tropical medicine and hygiene. He has a degree in PGPE. He also has a degree in American Academy of HIV Medicine Specialist, and he's a fellow of the Royal Society for Public Health. So in addition to this, he's the assistant professor in infectious diseases department of KMC Manipal, KMC College, Manipal, India. He's an infectious disease physician scientist with experience in clinical, epidemiological, and basic science research. His interests include in HIV, fungal infections, and tropical infections. Okay. He's a published author of 7 chapters in reputed national and international books. He's also published author of 108 peer-reviewed papers in national and international journals. It is our great privilege and honor to have you here, sir. Welcome, Dr. Nitin Gupta. Over to you, sir.
Thank you for the kind introduction. Is my screen visible?
Not yet.
Yes.
Yes, sir, your screen is visible now, sir.
Thank you so much. So I'll start my presentation. So I was given a topic, mycosis lurking in the shadows. So what I'm going to concentrate on is the identification of these clinical mycosis with potential outbreak potential. And I'm going to focus predominantly on the clinical diagnostic and management part and touch on the epidemiology. So because this is a huge topic, all I'll be able to do is give a bird's eye view. And so for the purpose of this discussion, I've divided fungal outbreaks into community acquired outbreaks and nosocomial outbreaks. The community acquired outbreaks can either be due to an occupational or recreational exposure, So what I've done is that with each of these subheadings, I'm going to take an opportunity to discuss one of these, the common fungi associated with that mode of outbreak occurrence. And so for occupational and recreational, I'm going to discuss Histoplasmosis and Cryptococcosis. In natural disaster or conflicts related outbreak, I'm going to discuss Mucormycosis. I'm going to predominantly concentrate on mucormycosis because this is the it outbreak right now, and the whole country has been dealing under this mucormycosis outbreak. And then we'll move on to the nosocomial outbreaks. For construction-related outbreaks, I'm going to discuss aspergillosis and some hospital-acquired outbreaks related to poor infection control. I'm going to discuss Candida. So in community-acquired outbreaks, there can be sometimes exposure to, for example, soil that is contaminated with bird or bat droppings, which has a higher propensity to grow histoplasmosis. And therefore, it has been seen that individuals who have been exposed to soil that is rich in bird or bat droppings, there has been a tendency to cause histoplasmosis. But the thing is that the disease is usually self-limiting, and it manifests more in compromised patients. But it should be noted that there have been several reports of histoplasmosis all across the country in immunocompetent individuals as well. Because often this happens when there are a group of people visiting a cave which has a high bat population, and there is a lot of exposure. So these outbreaks can be point source outbreaks. And also what happens is because the fungus is in the soil, any activity that disrupts the soil by like construction, bulldozing or soil tilling, this will lead to people getting exposed to this fungus. It has been, there have been studies which have shown that Cryptococcus is also found in soil that is rich in pigeon excreta, but the association is less clear. No, not all patients. In fact, most patients of Cryptococcosis that we see do not give any clear history of exposure. That association is more with histoplasmosis, but I'll discuss both histoplasmosis and Cryptococcosis here. So I've just put up this photo of a cave in Switzerland. The idea is to introduce the term spelunking. This is something that these are basically cave explorers, people who like to visit caves. And during this hobby, there is a possibility of exposure to certain fungi. I'm going to discuss this small case. This was a 58-year-old male spelunker who had low-grade intermittent fever for 9 months. He had erythematous papules over face and trunk for last 1 month. You can see these molluscum contagiosum like lesions. Some are papular, some are umbilicated. And he also had pancytopenia. Incidentally, he was found to be positive for HIV, and you can see that the adrenal glands are enlarged. So what you'll see here is the bone marrow aspirate of this patient, and also the skin biopsy showed similar lesions. So what you can see here is the yeast forms of Histoplasma capsulatum. So Histoplasma capsulatum, while capsulatum is what we see in India and in classically the Ohio River Valley in USA, but there is another Histoplasma subspecies duboisi, which is seen in Africa. It's just that morphologically they are a little larger. Like I said, people who are involved in activities that can lead to higher exposure, plus people who are immunosuppressed are at increased risk factor. Traditionally, histoplasmosis have been described in India along the Ganga and Yamuna River Valley. The first case was in West Bengal, but traditionally a lot of these cases were here. This has led to a false assumption that histoplasmosis is found only near the river banks. That's not true. It might be that cases might be a little higher there, but it has been reported all across the country. When I moved in to Karnataka, I was told that histoplasmosis is not there in Karnataka. But again, this is a review from Mycopathology, where all the published cases of histoplasmosis was charted out, and there were cases from Karnataka. And in my short span of time here, I've recently come across 2 cases of histoplasmosis here as well. Because the exposure is through inhalation route, the primary manifestation is pulmonary, but one should note that most immunocompetent individuals will have a small nodule which will go unnoticed, and it is often common to see detect histoplasmosis on a CT scan which you have done for probably some other reason. But in those some immunocompetent and otherwise immunosuppressed individuals, the primary presentation is the acute or chronic pulmonary disease, and with higher immunosuppression, there's a tendency to cause progressive disseminated histoplasmosis, in which, like I have shown in this case as well, there is bone marrow involvement, there is skin involvement, there can be bilateral adrenal involvement as well. One should note that this skin is a very good sample for demonstrating histoplasmosis. If you don't have skin lesions, then bone marrow is the next best sample. The diagnosis, like I said, is based primarily in dissociated settings by histopathology. So you see the yeast forms of histoplasmosis on GIMSA. You see the intracellular narrow base budding and granuloma formations in histopathology. Fungal culture can be done either through bone or bone marrow blood culture, and ideally, you have to demonstrate the mold form that grows in culture to yeast to show that it is histoplasmosis. But again, this is a very time-consuming, and often in source limited settings, these cultures might not be available. Serology is of limited use for histoplasmosis, especially in our settings. Most of our patients have negative serology. Antigen is an immensely useful test for detection of progressive disseminated histoplasmosis. The sensitivity is very high in progressive disseminated histoplasmosis. In limited histoplasmosis, it might not be that high. The only problem is this is an expensive test. But the thing is that because a lot of these patients go undiagnosed and they're diagnosed with non-specific things like HLH, it makes sense to invest in such a test. What I've shown here is the tuberculate macroconidia of histoplasmosis. This is the mold form that grows in the culture. So the treatment, again, for mild disease, you just have to give itraconazole for a small duration. For chronic diseases, the duration have to be longer. In retroviral disease, the common form that we usually see is the progressive disseminated histoplasmosis, like the case I discussed. So here you start liposomal amphotericin B for a period of 7 to 14 days, followed by prolonged itraconazole. There was a recent trial there, because there's a tendency in physicians that the higher drug or the more expensive drug is better, and so therefore a lot of people wanted to replace itraconazole with voriconazole, and there was a small trial that compared itraconazole versus voriconazole for suppressive therapy of histoplasmosis, and the trial failed miserably. And so the treatment, the current standard of care remains itraconazole. This is another case, 40-year-old pigeon keeper, patient had fever with headache for 3 months, found to be retroviral disease, CD4 is low. The first thing first is that if any patient of retroviral disease with low CD4 count comes with headache, even if the patient does not have any meningism, it makes sense to evaluate for cryptococcal meningitis. A lumbar puncture is a must. Even if you're not able to do a lumbar puncture, you should evaluate for serum cryptococcal antigen. I did show this laughing dove here to impress that there might be a possible association, but very often, like I said, you might not get any history that the patient had exposure to pigeon droppings. What you usually see in lumbar puncture is a kind of a paucicellular response. The thing is that because these patients are often immunosuppressed, there might not be a good immune response, and therefore the cells are not usually high, and this helps you in differentiating with other causes of chronic meningitis where you can get a cellular response. Sugar is not that low, protein is not that high. Whatever cells you will get is mostly lymphocytic. But the thing is that you will have to keep in mind that if your cellular response is high, it can still be cryptococcosis, but then the prognosis is better, because that suggests that the immune response is good. What you see here is in the lowermost panel of the slide, you see the India stain, which is a cheap test for cryptococcosis. It works well for HIV patients, but we often diagnose cryptococcosis in immunocompetent individuals as well. There it has a very poor sensitivity, and the gold standard remains a cryptococcal antigen test, which is not very expensive, and now lateral flow assays are also available. This is a mucicarbine stain of one of the patients. This was from a pulmonary sample, but I've just used this for representation purposes. What you can see is that India has a very high load of cryptococcosis, and I think most people will agree that this is one of the most common causes of CNS manifestation in HIV patients that we see in tertiary care hospitals. Diagnosis, like I said, the first thing probably that you will see in CSF is elevated pressures. So often when you do CSF, you don't do manometry. It makes perfect sense to do in patients who are suspecting cryptococcosis, because one of the treatment targets would be to reduce the elevated pressures. There have been some old trials which have shown that mannitol and acetozolamide does not work in cryptococcosis, and therefore the only way to manage high pressures in CNS cryptococcosis is by therapeutic lumbar puncture. Often we are required to do it almost every day or alternate days. Other than that, like I said, mild or elevated protein and low to normal glucose, and very low cellular response with lymphocytosis. The gold standard is cryptococcal antigen. I cannot impress more that serum cryptococcal antigen is a very, very useful test for cryptococcal meningitis. But the thing is that there is no point of repeating it multiple times. There are lateral flow assays available where you possibly not able to do a titer, but in latest agglutination test you can do a titer, but its serial cryptococcal antigen test might not be very useful in prognosticating. India has poor sensitivity, especially in immunocompetent individuals. Culture is what is recommended in IDSA guidelines, but WHO guidelines says that in this limiting setting, it's often difficult to do culture, although it's useful. So the IDSA guideline says that you start with induction therapy and continue until your CSF is sterile, but the new WHO guideline says that you need not do culture or repeat culture in resource limited settings. Again, for people who follow up to date for management of cryptococcosis, you will see that the regimen is liposomal amphotericin B with fluconazole for 2 weeks. But this is based on a very old IDSA guideline. But recently there was a trial in Africa where they showed the superiority of 1 week liposomal amphotericin with flucytosine, based on which WHO in 2018 has changed its recommendation. So it's very important and helpful, because in that trial they used deoxycholate, and for most of our patients who are not able to afford liposomal amphotericin B, we use deoxycholate only. So the current regimen now is use of 1 week of amphotericin B with flucytosine, and followed by 1 week of fluconazole at a very high dose. Unfortunately, the recommendations were not weight-based, and often a lot of our patients who have cryptococcosis have a very, very poor low BMI, but we are forced to use this dose only. But as of now, I've not seen a lot of liver function derangements because of high dose of fluconazole. Alternatively, some of our patients who are not able to afford admissions, or in government hospitals where the bed crunches are there, you can use a regimen of all oral regimen where you can use fluconazole and flucytosine for 2 weeks. This works really well. What I can tell is that the common things that were used, that amphotericin B and fluconazole, were shown to have a higher mortality as compared to both these regimens. Then you move on to a consolidation phase of fluconazole 800 mg. Again, this is a change from the previous guideline. You have to use this for 800 mg only for 8 weeks, and then maintenance based on the recovery and viral load for at least about a year of 200 mg of fluconazole. Some important points about cryptococcosis is that adjunctive corticosteroid therapy is not recommended, because a lot of these patients are suspected to have TB meningitis as well, where you want to give steroids. But if you are definitely sure that this is not TB meningitis and this is protocol meningitis, steroids are associated with increased mortality. The other thing is that in general, in retroviral disease patient, the dictum is to start ART as early as possible, even though there is a risk of iris, but you start ART as early as possible so that the patient does not die of a new opportunistic infection because of the lower CD4 count. But for cryptococcal meningitis, there is enough evidence which shows that early ART leads to higher mortality. So this is probably one of those conditions where you have to delay ART by 4 to 6 weeks. Like I said before, you might require therapeutic lumbar puncture, and in settings where you can do it, you can do LP and you can decide your induction therapy based on that. You can demonstrate sterility in follow-up cultures, but usually in low middle income countries, it's not required and we don't go for that. So now we move on to the next part of the community-acquired outbreaks. It could be natural disaster or human conflicts. We'll go to a little bit of history. So in Colombia, South America, there was a volcanic eruption in 1985, which was associated with 38 injured patients who had necrotizing lesions. 8 of these patients, Rhizopus oryzae, was isolated. So when we go talk about epidemiology of mucormycosis in India, it has been seen that quite of these patients are cutaneous mucormycosis post road traffic accidents. In India, although the species are a little different, that will come to later, but yeah, post volcanic eruption, there were these cases. In Missouri, in USA, there were 13 cases of hypophysomyces post a tornado. What happened was that in the path of the tornado, there were a lot of shreds of stuff that were flowing around in the tornado, and that led to penetrating trauma, and which led to increased number of wounds and increased number of infection with the spores that were displaced during the tornado from the soil, and that led to cutaneous mucormycosis cases. Incidentally, apophyzomyces is the second most commonest cause of mucormycosis in India, very commonly associated with post-road traffic accident. In the Indian Ocean tsunami, also there were several isolated case reports of soft tissue infection due to mucormycosis. So the point that I want to highlight here is that apart from the causes of mucormycosis or the acquisition of mucormycosis that we're going to discuss, this is one of the common things, especially in India, where you would see mucormycosis post, a large road traffic accident or an earthquake or anything that leads to displacement of soil and skin soft tissue injury. Usually, these patients have very rapidly expanding large necrotizing lesion, and apophyomyces and succinia are the commonest ones in India. The other thing that is interesting is that there was a large series that was reported from Chandigarh where they found that in local villages where they were reusing injections, post-injection, like improper intramuscular injection, there were reports of mucormycosis. So these are all you can see either on deltoid or on gluteal region. There were a lot of these injections. Most of the time, these injections are given for placebo purposes, these multivitamin injections, and this is a problem. So I'll move on to the next case. This is a 35-year-old patient with diabetes mellitus, mild COVID, and I'm sure everybody has seen a lot of these mild COVID patients were prescribed steroids for high dose of steroids for a long duration, and then they presented with this right-sided facial swelling, along with this palatal perforation. On CT, you saw pansinusitis. One thing that I'll say, one thing that I commonly seen happen is that people take swabs and it grows aspergillus or something else, and then people want to treat with voriconazole. So the thing is that one, if there is high risk factors. Second, if there is a rapidly progressing lesion, until you are sure, treat it as mucormycosis. So this patient also grew aspergillus in the culture when they did a superficial swab, but when they insisted and got a debridement done, it was full of aseptate hyphae suggestive of mucormycosis. The other thing is that you can have these florid lesions, and when you see these necrosis, there is no point thinking of anything else as a possible diagnosis. This is definitely mucus. So it can either go up in the eyes and brain, or it can go down and cause fatal perforation. But either ways, keep a very high suspicion of mucormycosis. There are risk factors such as diabetes, steroid intake, and rapidly progressing lesions. In this patient also, what you can see is that these three things were seen on a point of care testing. This was a small swab that we took, and we saw these three things. This is so you will find stuffs like, or this is a spore of Alternaria alternata, which was again a possible colonizer. But again, there is a tendency to go and try to treat fancy things. And this was the Demodex Nitte, which is again probably a common cell here. But the thing that we saw here is the septate hyphae. So even if there is a sea of septate hyphae and something else, if you find one aseptate hyphae, that is enough to make a diagnosis of mucormycosis, and that patient requires and deserves treatment for mucormycosis. So because this is the thing that we are dealing commonly now, post the COVID thing, I think whatever we saw before, the numbers have increased by a huge margin. But just want to impress that it's not that mucormycosis was uncommon before COVID. If you see the estimated incidence per million cases, India and Pakistan had a very high number of cases compared to the Western world. The other thing is that if you see in India, the epidemiology is a little different in the sense that our species are a little different. We have a high number of hypophysomesis and probably apophysomesis and lithemia a little bit. The other thing is that in India we have rhino-orbital cutaneous, rhino-orbital cerebral mucormycosis as the most common presentation of mucormycosis, whereas in Western world, where mucormycosis is mostly associated with transplant patients, it is pulmonary mucormycosis. The classical risk factors that we have always seen is patients coming with diabetic ketoacidosis. Both diabetes and acidosis are independent risk factors for mucormycosis. But what we see now in this COVID era is two things playing simultaneously. One is the uncontrolled diabetes mellitus. One is that there is an unmasking of diabetes because a lot of patients do not visit hospitals before and are diagnosed with diabetes at the first point of time. Second is with the high dose steroids, we push the sugar levels to a higher level. I have been told by many people working in periphery that they consider 400, 500 levels of sugar to be okay for COVID patients. The other thing is that steroid therapy in itself is a risk factor for mucormycosis. So India as such has a very high incidence of prevalence of diabetes mellitus, and also we had a higher number of mucormycosis cases before, like I discussed. And most of these cases of rhino-orbital cerebral mucormycosis are diabetic. Very rare to find cases who are not diabetic. And there was a study by Prakash et al. Where they found out that the diagnosis of mucormycosis unmasked diabetes mellitus in a good 20-40% of the patients. So how diabetic mellitus predisposes to mucormycosis is that they cause an increased expression of GRP78 on blood vessel on endothelium, and this leads to increased mucor attachment and invasion. If you see, most reports from India have reported very high incidence of diabetes and diabetic ketoacidosis in patients with mucormycosis. Just one thing, I've not kept a lot of slides, but in the last 2 or 3 months, we have seen like 6 or 7 cases of pulmonary mucormycosis post-COVID, which was not as common in non-malignancy or non-transplant patients, but this has increased as well. So keep a high index of suspicion for patients who are presenting with suspected pulmonary mucormycosis as well. So the things that might have culminated towards increased incidence of mucormycosis in patients who have recovered from COVID is probably you know that diabetes mellitus being an independent risk factor for severe COVID, and therefore patients who are ill are more likely to have diabetes, and therefore they have higher risk for mucormycosis. There was a lot of steroid use, and there is no doubt that steroid is probably one of the most useful drugs for COVID-19. But the thing is that it's a double-edged sword, and when you use it inappropriately at inappropriate doses, or use it for patients who do not require it, that is going to be very, very risky. The other thing is that there were some studies that showed that there is increased availability of free iron, and iron is a pro-inflammatory thing, and there are studies that have shown that increased free iron increases the invasion of mucormycosis. Also, we have all heard about NLR ratio, and there is decreased lymphopenia during COVID, and that as an independent risk factor for severe disease. So lymphopenia leads to decreased CD4, CD8, and therefore there's a dysregulated immune response. And there are now recent studies that have shown that there are receptors of SARS-CoV on pancreatic cells that are actually causing dysglycemia independent of the critical illness state or steroid use in these patients. Also, higher levels of GRP78 that I just explained, that is a receptor for mucormycosis. In this slide, I just want to point out that in patients with mucormycosis, in the late COVID-associated mucormycosis, in this multi-center study, they found out that only 33% of steroid usage was appropriate. This is a big study of mucormycosis. The consortium for ophthalmologists published this study, and they found out that a lot of these patients did not require oxygen. 57% did not. 57% required oxygen, and 43% did not require oxygen. But even then, 87% received steroids. So there is a definite inappropriate use. And a lot of patients were given steroids for prolonged duration, which, as per the last evidence of recovery trial, it was for 10 days. And diabetes mellitus was present in 78% of the patients. In retrospective study, there is a tendency to underreport diabetes mellitus. At least in our experience, we've seen most patients had one diabetes mellitus and second use of steroids. And the mean around 2 to 3 weeks is the time when patients develop mucormycosis. The mortality has been surprisingly low, probably because there was a lot of media attention, and so therefore patients presented early, and therefore hospitals were able to intervene. We have had surprisingly a very good discharge and recovery rate for mucormycosis. The diagnosis, like I stressed before, that microscopy is very, very helpful. Broad aseptate ribbon-like hyphae with wide range or right angle branching at regular intervals. It is very important to keep all these points in mind while you are diagnosing mucormycosis. The other thing is that calcoflow stain is helpful because it helps you in easy and early diagnosis, especially for non-experienced microscopists. One thing that is very important to remember is that culture is difficult for mucormycosis. The hyphae is aseptate, and so the nuclei is like floating around in the hyphae. And so once you break and cut these tissues for processing, these nuclei fall down, and then the culture becomes very difficult. Usually, the culture with mucormycosis it grows fast, but may not be positive in all cases, and therefore you have to rely very, very heavily on early microscopic diagnosis. In good centers, we get a lot of culture reports, but our treatment is primarily decided based on the KOH stain. Swabs are usually not recommended. And like I said, keep in mind that you can have a lot of fancy fungi that you can see or grow in your non-representative tissue sample. But till the time you have a good surgical specimen that does not show any mucormycosis, don't rule it out. There are now some PCR-based tests as well that are available, but again, its availability is a little difficult in resource limited settings. So CT scan is very helpful early on to, at least for the surgeons, to decide to what extent do they want to do the debridement, and they are very good for bone erosions. But in general, MRI is better to see the infiltration of periantral fat plane, and soft tissue vessels are better visualized. And there are some signs which are supposed to be better. Thickening and lateral displacement of the medial rectus muscles are supposed to be specific, and you see hypo on T2 and ISO to hypo on T1. The thing is that if you have a patient with a significant risk factor, with a matching clinical history, and CT or MRI showing pansinusitis, keep mucor as a first possibility and try to start your medical therapy as early as possible. The principles of treatment is that you have to control underlying disease. One is that you definitely have to prevent hyperglycemia during your hospital stay. We keep a goal of around 140 to 180 sugars in patients who are admitted with us. And the other thing is that once you're diagnosed with mucormycosis, try and reduce the sugar levels. Surgical debridement is very, very important. This is actually the mainstay of therapy. If you do not debride the necrotic tissues, the fungal, the antifungals are not going to reach where you want them to reach. And the third thing is specific antifungal therapy. So because a lot of centers sometimes delay surgery or biopsy, thereby delaying diagnosis, and there is a tendency to not start antifungals till the time a definite diagnosis is made. And so I just want to show a study where delaying by more than 6 days in initiation of antifungals resulted in twice the mortality. So in general, in those patients where they are suspecting mucormycosis, we start amphotericin on day 1. Surgical management, like I said, has good advantages because antifungals are not able to reach the necrosed part, and so debridement of necrotic tissue is very, very critical. And surgical management is independently favorable for good outcomes, and mortality is definitely lower in those with surgery. But if you wait till CNS involvement, there was a study that did not show that surgery helps. So once patients have CNS involvement, that is a time where it becomes very, very difficult to save the patient. So amphotericin B, now there is a global guideline that says 10 mg per kg liposomal amphotericin B, but in most private hospitals, it's absolutely difficult for patients to afford such a high price. And during the pandemic, the prices were even higher, and it becomes very, very difficult to treat with liposomal amphotericin B. So although the guidelines said that do not use deoxycholic amphotericin B for our country, it becomes very difficult. We treated all our patients with deoxycholic amphotericin B. There is no option that we had, and the results were quite favorable. Yeah, you do have AKI, you do have hyperkalemia, but you are very aggressive with medical management. It's okay. Most of the times AKI is reversible. There is a tendency to stop amphotericin B the moment creatinine rises a little bit. There are again no studies as such, but I would advise against it. What we usually do is we keep a higher threshold of creatinine. We wait till 2.5 and then stop it for a day or two and let it come back and then again start. But this, what is recommended by the global guideline is use 5 to 10 mg per kg if there is no CNS involvement. If there is CNS involvement, use 10 mg per kg. But again, this is an expert-based recommendation. If I'll just show you one, there are no trials in mucormycosis. There are no RCTs in mucormycosis. That's very unfortunate. Just this one open single-arm study that used 10 mg per kg of liposomal amphotericin B, and there was a higher incidence of nephrotoxicity in these patients. So the point is that higher incidence, higher dose of amphotericin B probably is required for those where we want CNS penetration, but we know that already the prognosis is poor with patients with CNS involvement. It's a very difficult thing. Unless and until we have trials, it will be difficult to definitely say whether this is a good enough recommendation or not. As of now, for our routine practical purposes, we use deoxycholic amphotericin B at 1 to 1.5 mg per kg. There were some issues with availability of amphotericin B for a while, and at that point of time, there was a lot of marketing for use of posaconazole and isavuconazole. The short statement that I would like to make is that they are not a good enough alternative for amphotericin B. You have to try and get amphotericin B first. If there is no option, then there's nothing we can do. But posaconazole serves well for step-down therapy. The studies that are there with posaconazole are predominantly retrospective case series. The studies are therefore not that good, but because a lot of these patients require long-term suppression, therefore posaconazole is better. But just keep in mind that posaconazole has poor CNS penetration. These were the two studies that were there, some small review articles or case reports. Just one thing that I would like to point out is that for posaconazole, it is better to give tablet than the suspension form, because the absorption of suspension form is a little erratic. Isavuconazole again is a really expensive drug, but it has a good CNS penetration compared to posaconazole, and this again can be used for as an alternative for suppression therapy. Again, as a primary drug, the data is less. There was this vital study that showed that non-inferior outcomes with isavuconazole, but this was not a randomized controlled trial. This was a retrospective comparison study. Combination, as such, there is no definite evidence to combine drugs, and just makes the regimen more expensive. And in my personal opinion, feels it makes more sense to give amphotericin B initially as much as possible, and then use whatever money you have to buy the suppression therapy and prolong it as much as possible, instead of giving combination therapy where the evidence is not there. There were some reports of initially that iron chelation therapy works, but DEFEAT trial did not show any benefit. Again, there are problems with the DEFEAT trial as well, but as of now, there isn't enough evidence that deferazole works. So this is what we commonly use for our center. Is we try to give amphotericin B as much as possible for 3 to 6 weeks, even longer sometimes, but we try to give at least for this much duration. And then if the patient has improved clinically and radiologically, we shift them to posaconazole or isavuconazole. We try to put them on tablet. Posaconazole is a little cheaper, so that is what we do. If there is no clinical radiological improvement, you can consider all these things, but again, for most of our patients, it becomes very difficult. They are not able to afford these regimens. So next, I'll move on to the nosocomial outbreaks, and I think this is a common scene that you can see here in most university hospitals all across the country. There is some other construction going on. This is something which is inevitable, but the thing is that we have to know what are the consequences of construction around hospital facility. So there are lots and lots of reports of healthcare associated aspergillosis outbreaks in hospitals where construction is going on, because this leads to dispersal of fungal spores, and it's more commonly with aspergillus. Like I said, mucor post-natural disasters also happen. Similar way, mucor can also happen, but it's more commonly seen with aspergillus. Most common site is pulmonary. And the thing is that most of these patients who get pulmonary aspergillosis in, possibly related to construction exposure, are in patients with risk factors for mucormycosis. So this was a big review of construction-related aspergillosis, and you will see that most number of patients were patients with hematological malignancy or patients who are on other immunosuppression, and there was a high amount of mortality. Why I'm pointing this out is that when you are doing a construction facility, just keep in mind that whether your vulnerable patients are housed near that construction facility. This is these things might be helpful in prevention of fungal infections, and you have to inform your infection control team that you are undertaking a construction facilities and take their inputs as to how to conduct best and cordon off that area. You have to conduct an infection control risk assessment as to who are the people who are exposed because of this construction facility construction work, and can we do something to house them somewhere else, and try and put barriers so that you are able to suppress the airborne dissemination of spores, and maintain surveillance if there are any filamentous fungal infections, aspergillosis, cases happening in hospital, see if they can be attributed to the construction, and try and see whether you can do anything better to decrease any further cases. And also regular visits by infection control team is required to see whether whatever regulations or advice that they've given are being complied to or not. This is a small case that I will take. This is a 60-year-old male patient with AML, induction chemotherapy, consolidation therapy for 5 days, uneventful hospital course, came back day 6 post chemotherapy with what you can see. And this is very clearly invasive aspergillosis. You know, this is the left one is the CT halo sign, and this is the crescent sign. This sign is seen commonly in patients who are neutropenic only, and AML is a definite risk factor. Most of the patients are put on prophylaxis for aspergillosis before starting induction chemotherapy, but then these patients may have invasive aspergillosis. So like I said, neutropenia is a big risk factor. Keep in mind that HIV is not that big a risk factor because HIV leads to decrease in lymphocyte counts when neutrophil count is usually okay, which is the primary, neutrophil is the primary defense barrier to aspergillosis. And therefore, you have to especially keep in mind when you have a neutropenic patient and other transplant recipients and all. Just one thing that I would like to add here is that in the recent few years, there have been a lot of cases of invasive aspergillosis post severe influenza, and this is something that you should keep in mind. There are different criteria. Just to make one point put across is that these classic signs of CT halo and air crescent sign are seen in patients who are neutropenic and then are slowly recovering. But in patients post COPD or post influenza or in other ICU critically ill patients where aspergillosis is also seen, or in post-COVID patients, you will not find these signs, and this makes it very difficult because most of the time the diagnosis of aspergillosis is made on clinical radiological grounds. Galactomannan, unfortunately, in most places where I have worked, has not been very helpful. Most of the patients that we send galactomannan for can come out to a positive, and therefore it becomes very difficult to make a diagnosis. So I'm not going into the details of the guidelines, but anyway, so just to keep in mind that the radiology might not be very classical in routine critical ill patients, and you see like septate hyphae branching at 45 degrees in patients with aspergillosis, but the same thing can be true for any other hyaline mold. So just keep in mind, galactomannan assay is classically told to be very specific, but again, it is a little difficult to interpret in some of the settings. And the sensitivity is lesser for non-neutropenic adults because the galactomannan is supposed to be released from the tips of the ends of actively growing hyphae. If you have neutrophils in your system, neutrophils will try to remove aspergillin, the galactomannan from the blood, and therefore classically in neutropenic patients, galactomannan assay is supposed to have more sensitivity. But in our centers where the work has had a very high positivity, galactomannan often makes it very difficult to diagnose. So the treatment for aspergillosis is definitely voriconazole. There was a big trial that compared voriconazole versus amphotericin B and showed that voriconazole works better. So also voriconazole has oral formulations available, so you can, as and when the patient is able to take orally, you can shift the patient to oral voriconazole, and it is cheaper as well. Like I said, that this is a classic halo sign and the crescent sign for aspergillosis, but in ICU patients, you will not find very commonly these same signs. And in like high burden ICUs, we have had aspergillosis cases up to 10 to 14% in all critical ill patients as well. This is one of the outbreaks in Louisiana where they reported cutaneous mucormycosis post exposure to contaminated linens, and when they did an outbreak investigation, they found out that these clean linens were kept in a laundry facility in bins which were contaminated with rhizopus. So good infection control is very important to prevent these nosocomial infections. Then there are these classical reports in USA of patients receiving contaminated medicine. So one of these was a patient undergoing vitrectomy, received contaminated injections into eyes, and this led to infection with Fusarium and bipolaris in a lot of patients. Similarly, there was another outbreak where patients received injection with haloperidnisolone that lead to CNS meningitis with exerohyalin rostatin. So, but these are fairly uncommon. To my knowledge, there are no reports as such of them in India. I'll take up the last case to highlight the problems of contaminated medical devices that can lead to fungal infection outbreaks. So this was an 80-year-old male, which is rescue of Boerhaave syndrome. I'm sure you know Boerhaave syndrome is after intensive retching in old individuals, there can be esophageal rupture, and the patient was admitted in ICU, and then they did a transcytal repair with mediastinal drain. Patient was put on total parenteral nutrition to central line, and on day 4 had a new onset fever. We started empiric antibiotics as we most often do, and then central and peripheral paired culture showed a growth. You can see the candida growing here, and you can see that there is an exit site infection here. And so you saw similar fever in other patients. This should alert you an outbreak related to candida. One thing that I'll point out here is that there are several risk factors of candida in these patients. Esophageal rupture is an independent risk factor for candida. ICU admission is a risk factor for candida. Any GI surgery is a risk factor for candida. Total parenteral nutrition is a risk factor for candida. Empiric antibiotic use is a risk factor for candida, and central line is a risk factor for candida. There were so many risk factors for candida in this patient, and therefore, when you have these patients with risk factors for candida and anticipating candidiasis, good infection control is very, very important. And these are the risk factors that I've already shown. If you see in Western literature, Candida albicans is more common, but in India, non-albicans Candida is commoner, and you see Candida tropicalis. The other thing is that we have incidence of 6.5 cases per thousand. This is based on a multi-centric ICU-based study done by Dr. Chakravarti from PGI. And so what they saw was that younger patients were more commonly found to have candidiasis in the Indian cohort, and they had a lower Apache score, and infection was early compared to studies in Europe and US. But the good thing was the azole resistance was not as high. Even with glabrata, which is traditionally thought to be having high resistance for azole, in at least Indian isolates, the resistance to azole is lesser. But the thing is that mortality with Candida is still high. So although classically Candida is supposed to be resistant to fluconazole, but in India there may be isolates that are not resistant to fluconazole. Parapsilosis is associated more with invading devices, and although the therapy remains as the kind of candidates as the primary choice, but it has increased in MIC. These are some special things that are related to India that I'm just alluding to. Cruzii is definitely resistant to fluconazole, and Lucitan is resistant to amphotericin B. But the thing that I want to highlight is Candida auris, and there has been several outbreaks of Candida auris. It's a really dreadful pathogen, and once it comes in ICU, it becomes a nightmare for infection control people. So the incidence of Candida auris in all the cases is around 5.3%. It is associated with high mortality. It is associated with a huge nosocomial spread, and therefore you need to definitely isolate the patients with Candida auris and do chlorhexidine waiting, and there are several infection control recommendations related to it. One thing that you have to keep in mind is that identification with traditional phenotypic and some automated systems with Vitek might be faulty, and therefore Malditop is what is recommended for identifying Candida auris, and they have a high resistance to antifungals. Recently, I was discussing a case where the isolate of Candida auris was resistant to everything, and it becomes a nightmare for us to decide on therapy, and it has a very high mortality rate. The diagnosis of Candida is through blood culture techniques. Just keep in mind that blood culture might not be sensitive always. In those cases, things like beta-D-glucan is helpful, but again, it is very expensive and not available in many centers in the country. Identification of species can be done with some with Malditop very quickly, and it is very helpful to decide on empiric therapy. But in cases where you have a high, like for example, LAC patient, we started actually empiric candida therapy before we diagnosed candidiasis because the risk factors were so high. The therapy, the primary treatment recommendation is echinocandins, but again, a lot of our patients are not able to afford echinocandins. So in patients who are otherwise stable, we often give fluconazole, and we've seen good response with that. Once you show that the isolates are susceptible to fluconazole, you can switch from echinocandin to fluconazole. And you're supposed to do alternative blood cultures till the time you get a negative blood culture. The other thing that you have to keep in mind is that it's very, very important to do fundus examination, because oftentimes patients are critically ill and there are some candida endospermata that are there, and you do not diagnose it, makes it very difficult to decide treatment endpoints. And like with most central and associated bloodstream infection, you have to remove if there is any central. The treatment duration is usually 14 days after the first negative blood culture. With this, I will end my lecture. Thank you.
Thank you, sir. Am I audible? Yeah. Over to Dr. Shefali to pose the questions to the speaker.
Yes, thank you, sir. We have a few questions with us here. So do fungal secondary infections have genetic relation to viral infections? Example, cryptococcosis in HIV and mucormycosis in COVID.
Yeah, I don't think so. As of now, at least I am not aware of that. So most of the time with HIV, what happens is, see, HIV, the fungal infections are not like it will happen only in HIV. They happen in post-transplant recipients. They happen in malignancy, wherever there is immunosuppression. So it's more to do with immunosuppression. And like I discussed with regards to COVID, the occurrence of cases of mucormycosis was not. There are some studies now that say that possibly there might be some indirect relation that directly COVID is causing, but most of it is iatrogenic. It's use of steroids. There are some studies that are saying that probably there is increased expression of ACE2 receptor in pancreas, and COVID is directly leading to mucormycosis. But as of now, we don't have substantial evidence for that.
Okay, so next question. So do we have any invasive aspergillosis varieties in COVID-19? Are they seen? Are we missing their diagnosis?
Yes, there we have a lot of cases of COVID-associated pulmonary aspergillosis. Often the problem is that the diagnosis of this becomes difficult. So if you see the criteria for COVID-associated pulmonary aspergillosis that was given by a consensus group that said that if you have suspicion of aspergillosis and if you have galactomannan positive, you make a diagnosis of COVID-associated pulmonary aspergillosis. But again, like I said, that in some centers where galactomannan is used, there is a very high degree of positivity. It becomes very difficult. What we routinely use is the Blott's criteria for diagnosis of invasive aspergillosis. It is the criteria that was given in 2012, and the entry criteria for this Blott's criteria is that you have to have a culture positivity. So if we have typical radiological lesions that suggest invasive aspergillosis, or if we have culture positivity for invasive aspergillosis, along with supportive findings like galactomannan, we do treat with voriconazole, and yeah, it is common.
Okay. So as you mentioned, the biochemical markers in diagnosing these, what is the role of beta-D-glucan assay in patients who are already on beta-lactam antibiotics in the ICU setting, sir?
So I think the question is about galactomannan.
Yes.
So there was this before 2006, there were some formulations of epicelline tazobactam that Pfizer was making, which had some impurities that could lead to false positive galactomannan. But since then, they have revised their formulation. And so the newer brands of medicine that are available are unlikely to cause this false positivity. But again, what I suspect is that there is a high degree of false positive in galactomannan in our settings. Whether the reason is use of antibiotics or any other products that we are using, I'm not sure. But yes, there is often a lot of false positivity in galactomannan.
So what are the most common secondary infections due to fungus in patients with severe COVID-19 on immunosuppressants? How do we suspect clinically or biochemically for a fungal infection, sir?
Yeah, so I think the whole lecture was about how to suspect these fungal infections. So all as Candida, Aspergillus, and Mucor, all three are commonly seen. Mucor is by far the commonest that we have seen this time, but we have had a lot of cases of Candida and Aspergillus. So like I've discussed, the risk factors of these other than COVID are a little different, and I've already discussed all these risk factors.
Why does mucormycosis grow commonly in the rhinosinus source? Is it a normal commensal of the nose and the paranasal sinuses?
Okay. So no, so this is not a commensal, but the thing is that we regularly inhale a lot of these spores, but our normal mucociliary mechanism is good enough to expel them out. They usually do not stay in there, at least not in the sinuses. But during this COVID, when your immunity falls down, when you don't have your neutrophils not acting properly, you have lymphopenia, neutrophils not acting properly because of this, and then you have increased expression of receptors that helps in mucus getting a foothold there. So whatever you inhale has a higher chance to cause disease. Mucor is ubiquitous everywhere. We have a very high degree percentage of proportion or burden of spores in all air all around us, but it's just that normal immune system is able to take care of it. But during COVID, because of several reasons that I explained, you have a higher propensity to not be able to fight these spores.
Then, sir, as you mentioned that after 6 weeks in mucormycosis treatment, we see the clinical and the radiological improvement. If there are no improving signs, for how long do we continue the antifungal treatment? And do we have to consider alternative antifungals or add-on treatments?
So like I said, that in general, we have a good response with most patients. When you don't have a response, we go back and see whether the patient requires another surgery. Because if the surgery is inadequate, if there is a lot of necrotic tissues left behind, no amount of antifungals are going to work. There is no fixed duration. Like I said, there are no trials in mucormycosis. Most of it is expert based. So what we do is based on clinical response. So in those cases where your amphotericin B is not able to, there is no the benefit, the clinical radiological improvement has not been enough. We go back, we see whether a repeat surgical debridement might be helpful. We go for a repeat surgical debridement, and then again continue amphotericin B till the time we are happy that we can put this patient on a suppressive oral posaconazole therapy basis.
And so for how long do we plan to continue the oral posaconazole treatment?
That is, again, there is no trial as such, but so what we usually do is try to give it for at least 6 months and then see that whether there is a good enough radiological resolution. But the problem is that in the pre-COVID era, there have been certain patients where I have stopped at 6 to 8 months and then they have come back later with recurrence. So there is no definite idea of what is the total doses of posaconazole, but it depends again on what was the initial surgical clearance, how much is the residual tissue, and how has been the radiological response. But we try and give at least for 6 months.
Okay, thank you so much, sir, for this wonderful talk. I am sure that all of us have learned a lot and all our doubts have been cleared. Over to Dr. Siddharth.
Thank you, Dr. Nitin Gupta and Dr. Shefali. We're running around 20 to 30 minutes ahead of our schedule, thanks to the lack of any glitches on the technical front. As we know that our department has conducted this CME in a hybrid fashion, and we had 7 posters and papers presented from colleges across the state. And 7 of the papers were presented in the competitive category and 7 posters in the competitive category as well. And I'm pleased to announce that I have with me the results of these presentations. Coming over to the poster presentation, the first position has been secured by Dr. Sanjana from Shema. She's a third year resident in the Department of Internal Medicine. And the title of her poster was Reactivation of Tuberculosis in a SARS-CoV-2 patient. The second position was secured by Dr. Amoga Siddha Jyoti from KVG Medical College, Sulia. She presented to us a case report of non-ST elevation MI secondary to COVID-19 infection. In the second category, that is oral presentation or paper presentation, the first position was secured by Dr. Athira Patolat from Yenapoya Medical College. The title of a presentation was an outbreak of carbapenemase producing Klebsiella pneumoniae in an intensive care unit of a tertiary care hospital in Bangalore. She secured the first position. And the second position in the paper category was secured by Dr. Srivatsa R. He is a final year resident from Shema. And the topic of his research paper was neutrophil lymphocyte ratio and platelet lymphocyte ratio as a predictor of mortality in severe COVID-19 cases. My heartiest congratulations to the winners. And since we have time with us, we have the opportunity of seeing what the winners have presented. So I'd like to introduce you to Dr. Sanjana. As I told, she's a final year resident in the Department of Internal Medicine. And she's the one who secured the first position in paper presentation. And I'd like her to present the paper that she presented yesterday. Sorry, the poster. Over to you, Dr. Sanjana.
Just ask Siddharth to enable sharing. Siddharth, can you enable sharing for Sanjana?
Sir, can you hear me, sir?
Make Sanjana the host.
Can you hear me, sir? Very good afternoon to everyone. I'm Dr. Sanjana, third year resident from K.S.HEGDE Hospital. Today I'll be presenting a case report titled Reactivation of Tuberculosis in SARS-CoV-2 patient under the guidance of Dr. Giridhar sir. We all know that more than 1.7 billion people, that is about 25% of the world population, is affected by TB. But not all, so out of which only 5 to 10% have the risk of acquiring the TB disease. And out of this acquired TB disease patients, only 5 to 15 one affected person can spread it to 5 to 15 other individuals. So according to WHO in 2019, about 10 million individuals became ill with TB and about 1.4 million died. So we all know that tuberculosis is endemic in our country, India, Pakistan, Bangladesh, and these are one of the worst affected countries of the world. It is caused by Mycobacterium tuberculosis bacteria. So according to the WHO World Health Assembly, the End TB strategy, its aim is to decrease the TB incidence by 80% and to decrease the TB death by 90%, and also to eliminate the catastrophic cost for TB affected household by 2030. So to encourage in this regard, every March 24th of year is celebrated as World Tuberculosis Day. So the 2021 theme was clock is ticking, which means that the world is running out of time to act on commitments to end the TB made by global leaders. So TB, along with the, in addition to TB, now we have the COVID pandemic, so which is caused by the SARS-CoV-2 virus, which belongs to the beta coronavirus family. This first had its appearance in the Wuhan seafood market in China in December 19, and which continues to spread around the world. So both the TB and SARS virus, both are respiratory infections. They have some of the they share some of the common features, that is, both are communicable diseases spread by droplet infection, and both of them have the same risk factors, that is malnutrition, poverty, tuberculosis, and immunosuppression. These are the factors which will increase the risk of acquiring TB or viral infection like SARS-CoV-2. So in the previous also there are studies like viral infections like influenza virus or SARS-CoV-1, which also belong to the beta coronavirus family. It causes transient immune suppression that will lead to reactivation of dormant bacterial infection. In 1918, the Spanish flu pandemic gave rise to pulmonary TB, increase in the pulmonary TB incidence. And SARS-CoV-1 and MERS, that is Middle East Respiratory Syndrome virus, infected patients were reported to develop pulmonary TB. So this case report I presenting is about a COVID patient who post-COVID recovery who presented with a TB infection. So this is the case report of my middle-aged man who had a history of SARS-CoV-2 infection, which was a mild disease 8 weeks ago, who presented to us now with a cough of 4 months duration and a low-grade fever of 2 months duration. So he had no prior history of tuberculosis. Given the current corona pandemic, so we thought it is COVID. So COVID RT-PCR was done. The swab test was negative. And he also had undergone an HR-CT thorax outside, which showed mediastinal lymphadenopathy. So, so on examination, his vitals were stable and systemic examination was also within normal limits. His HR-CT showed pretracheal, subcarinal, and prevascular stations lymphadenopathy, with the largest measuring 2 to 2 centimeter in the right paratracheal station. We went ahead and did a endobronchial ultrasound with transbronchial needle aspiration of this mediastinal lymph node. The picture of this has been put up on the case report, which shows granulomatous lymphadenitis with caseous necrosis favoring tuberculosis. So diagnosis of tuberculosis was made, and according to the National Tuberculosis Elimination Program, ATT was initiated. So after the ATT was initiated, he was discharged in a stable condition, and follow-up was done through frequent telephone calls, and he had a symptomatic relief after a month of initiation of ATT. So coming to the discussion. So coming to the discussion, there are several mechanisms why this post-COVID patients are increased risk of developing tuberculosis. So as we all know, during COVID infections, we tend to give patients immunosuppressive drugs like steroids and in other interleukin antagonist and other drugs which will decrease the immunity. So, so some of the other mechanisms, as we all know, is a cytokine storm. So following the viral infection, there can be an increase of type 1 interferon, desensitization to toll-like receptor ligands, but that will lead to defective myeloid cell recruitment, impaired natural killer cell response, and production of anti-inflammatory factors, including interleukin. So, and also there are one of the important case report in this regard is one by Pathak and Pathak et al. In their study have put forth an important mechanism why this COVID infection can cause reactivation in a reactivation of TB in a person. See, as our patient did not have any comorbidities, neither did he receive any steroids. So what was the reason for him to acquire this TB disease? So one of the hypotheses put is that there are once a person is infected with tuberculosis, these bacilli, they harbor in a special fraction of cells called CD271 bone marrow mesenchymal stem cells. These stem cells, they constitute a very rare fraction, that is 0.001%. So whenever that virus infection, the entry of the virus, when the in a latent TB, that is, all of us are infected with tuberculosis. So this Mycobacterium tuberculosis will harbor in this small fraction of CB27 bone marrow mesenchymal stem cells. So whenever there is an acute insult to the individual, like a simple viral infection, or now the pandemic, that is SARS-CoV infection, this mesenchymal stem cells harboring this tuberculosis bacilli will migrate to the lung lesion. That is the lung, where they will provide a conducible environment for the virus to survive and for the MTB also to survive. So what they do is these stem cells will convert them into something called an altruistic stem cells. These stem cells, unlike the other stem cells, these stem cells have an altruistic nature. That is, they'll cause the Mycobacterium tuberculosis to survive the hypoxic environment in the host, and will cause Mycobacterium tuberculosis to activate and cause transbronchial spread and hematogenous spread. So this article, that is Pathak et al., they concluded that the SARS-CoV-2 activates the stem cell mediated defense mechanism, which can reactivate tuberculosis. So through this case report, why is it important for us? So as we all physicians, we come across tuberculosis in every day. So India being an endemic country, tuberculosis is a very common disease, and in this COVID era, the detection of TB has drastically reduced because patients are not presenting with any symptoms, fearing testing positive for COVID. So the detection rate have come down, and they are staying at home, and they are spreading it to the community. Like as I said, one person infected can present it to 5 to 15 other people. So we physicians have to be careful about this. So patient who has been discharged and who comes for follow-up, if he has any respiratory symptoms, or there is persistence of respiratory symptoms, or there are new respiratory symptoms, one should always think of tuberculosis and get a sputum examination and treat ATT at the earliest. So I would conclude by saying that SARS-CoV-2 causes a temporary suppression of cellular immunity, which predispose patient to aggravated reactivation or new infection with TB. So in a country like India, with a high burden of TB and ongoing SARS pandemic, we should be vigilant in detecting TB in previous SARS patient who has persistent symptoms or a new ALS symptoms, and initiate ATT at the earliest. So as SARS-CoV-2 infection significantly dysregulates immune system, apart from TB, we should also be vigilant in detecting other infections like Mucormycosis, Candida, and Pseudomonas during this post-COVID phase, which can occur as co-infection. Thank you.
Thank you, Dr. Sanjana. And congratulations once again for securing the first position. I'm pleased to announce that we have with us now Dr. Anusha Rohit, who will be presenting the next lecture titled Era of Bacterial Outbreaks. I now request Dr. Aditi K., Associate Professor in the Department of Internal Medicine, SHEMA, to introduce us all to the speaker. Ma'am, please.
Good afternoon, ladies and gentlemen. Am I audible?
Yes, ma'am, you're audible.
Okay. I have the great privilege of introducing our next speaker for today, that is Dr. Anusha Rohit. Madam is the head of Department of Microbiology and Senior Consultant in Madras Medical Mission, Chennai. She's also the Chair for Infection Control Committee and Assessor for Standards for Medical Laboratory. Madam has completed her MBBS from Rajiv Gandhi University in 2006, completed a MD Microbiology from Manipal University in 2010, and we are proud that she was an alumnus and completed her PhD from Nitte University in 2019, and also completed a diploma from Royal College of Pathologists 2019. Madam has a number of awards and fellowships to her credit. The most recent one being from the Society for Healthcare Epidemiology of America for the commitment to advancing healthcare epidemiology, infection prevention, and other goals of SHIA in India. She also has a number of national and international presentations to her credit. She's also won a number of awards in these presentations. She has actively conducted several national and international workshops for betterment of microbiological studies and development. She has also participated and has keen interest in medical training and education and published books in this context in the subject of microbiology. She has a huge number of publications in both national and international peer-reviewed journals. And these are the publications of Madam in the last 3 years. Some of you must have also seen Madam in national television delivering certain interviews about COVID in the last year. We are very honored to have, for Madam to have accepted our request to be one of the speakers for today. Now I'd like to hand over the platform to Dr. Anusha Rohit for her talk on the era of bacterial outbreaks.
Thank you so much, Dr. Aditi. Let me just share my presentation. I hope you're all able to see the slides.
Yes, ma'am, the slides are visible.
Perfect, thank you. So at the outset, I really want to thank the Medicine Department of K.S.HEGDE Medical Academy, my alma mater. I did my MBBS there, I did my PhD there. I just love that institution for reasons beyond. And thank you for giving me this opportunity to be a part of this program. So I'll get right into the topic of bacterial outbreaks. And since we've spoken about outbreaks all through today, from the initial Dr. Yamuna's presentation on how to go about looking at an outbreak and on viruses, I unfortunately had to miss the mycology outbreak talk, but I listened to the previous speaker on the TB, and we are also seeing a lot of TB post-COVID that we are seeing, even say, for example, in pericardial tissue, etc. So outbreaks from bacteria are also very, very important. And I think, we've kind of forgotten many of the things that are, associated with bacterial outbreaks, especially after the advent of antimicrobials that came out in 1926 with Alexander Fleming. But let me take this opportunity to tell you that we are not far from the age where antibiotics are, going out of vogue because you have no longer sensitive bugs. I just returned from my HIC meeting and I had a huge argument with, one of our colleagues who was, questioning why we need to even do cultures, why is antibiotic resistance so important? And, I want to take this opportunity and this platform to explain to everybody that outbreaks due to bacteria can be devastating as much as that of viruses or of fungal, or for that matter, any other organism. So greetings from the Madras Medical Mission. So here are a list of the deadliest pandemics in history. So you have the smallpox, Spanish flu, etc. But let me focus on, I'll put this slide to explain this figure where you see the ones marked in red. You have the plague of Justinian, you have the bubonic plague or the Black Death, you had the Antonine plague, you also had the cholera outbreak, you had great plagues there. You see another word there, Kokolitzli. Of course, I had to Google it because I was also not aware of it until I was starting to prepare for this particular presentation, that it was supposedly because of salmonella, that outbreak. It was during the Mayan civilization. It was in the Mayan area, not during the Mayan civilization. It was in the Mayan civilization area. You also have the Russian typhus epidemic. We all remember how the World War I actually was half won because of the bacteria rather than, infecting all the troops rather than the fighting of the troops themselves, and so on and so forth. So when we're looking at all of this, if you kind of look at them by scale of size, the biggest one still holds to be Black Death or bubonic plague, caused by, all of you must be very well aware of, Yersinia pestis. You, of course, have smaller outbreaks here and there, which caused a lot of death. But we're all talking right now so much about the COVID virus, and you will see the COVID virus is right here, whereas we're talking about much bigger outbreaks that have happened in history. So going further ahead. So if I'm going to classify and take you through this particular talk through these, subheadings. I'm going to look at zoonosis, foodborne outbreaks, waterborne outbreaks, antimicrobial resistance outbreaks, which is right now currently the prolonged outbreak that we are all a part of, rather a pandemic that we are all a part of, and some toxin-based outbreaks. So I'm sure all of you learned this rhyme as a small kid. Ringa, ringa, roses, pocket full of posies, husha, husha, we all fall down. So little did I know till I was writing an essay when I was in 7th standard that this song was relating to plague. Ringa, ringa, roses, the buboes that were found, pocket full of posies, that was the herbs that were kept in people's pockets. Hasha, usha is the coughing and sneezing of people, and we all fall down is essentially the death of people that happened. So plague is something that essentially left no one to die. So it decimated Constantinople and spread like wildfire across Europe, Asia, North America, and Arabia, killing 30 to 50 million people during each outbreak. And in England, in fact, the London outbreak had people actually being locked into their houses and not allowed to come out. They would be left to die there. The sick were just left inside their houses. Please remember, this was all the pre-antibiotic era, so there were no antibiotics left. And there were no antibiotics present. The no antibiotics left is the present scenario, not then. And since then, there have been major outbreaks. So all of you know, excuse me, Yersinia pestis was not discovered, the organism was not discovered until 1894, during the 1894 pandemic, before which there were multiple pandemics and people did not know what was really causing this pandemic. It was discovered in Hong Kong by a French Pastorian bacteriologist called Alexander Yersin, which is why it's named Yersinia pestis. It is associated with fleas and rats, which is why it is called Yersinia pestis. So these are the main outbreaks that I'm talking about as far as plague is concerned. You have the Justinian plague, which started in Central Africa from 541, and it went on to 544, spreading across Egypt and Mediterranean. Whereas if you look at, this is all in CE era, you have the 1347 to 52 Black Death that started in Asia and Crimea and then went on to Europe and Russia. Then you have the 1665 to 1666, which was a great plague of London that started in London and spread across England and Europe. And the 1894 third pandemic that started in Yunnan, China, and really swept across the whole continent of Asia, Hong Kong and India, and then to the rest of the world. It was one of the largest outbreaks. And we are all wearing PPE right now. The picture on your right, you will see, is the PPE that was used by doctors during that time as they visited the houses of the patients with plague, and you will see mass graves being on the right side, where multiple people were just buried because they did not know how to take care. Now, if you think that plague was restricted to the less developed countries or the underdeveloped countries or the developing countries, you're sadly mistaken. If you're looking at the different outbreaks across the last 100 years in the United States, from 1900 to 2010, you will see there are large outbreaks, and you'll see clusters of them between 1965 to 2010. And that's not very far away. So this is not a new topic for us to see. Have we faced a plague in India? Yes, in India, there was an outbreak in 1994 that started in Bid district of Maharashtra and then went on to spread to Gujarat. And this is when I was asked to write an essay in my 7th standard. And that is when I discovered the rhyme Ringa Ringa Roses associated with plague. Subsequently, we had multiple cases of pneumonic plague in Delhi, Varanasi, Karnataka, and other states. And in toto, you have 876 cases with 54 deaths reported. As recent as 2002, an outbreak of pneumonic plague occurred in Himachal Pradesh with 16 cases and 4 deaths. So in 2004, again, you had a bubonic plague in Uttarkashi and Uttarakhand states with 8 cases and 3 deaths. So we are talking about very recent outbreaks. So with that, I'm going to go into the foodborne bacterial outbreaks. Again, a huge concept which we are still facing. Not only the Eastern world, the Western part of the world also is facing huge outbreaks as far as bacterial outbreaks are concerned. So here you see a picture of Mr. E. coli, who couldn't understand why his business was bad because he'd named his organization as E. coli vegetables. So first, let's understand, why do they occur? Where does contamination actually occur? Is it occurring during the phase of growing them? Is it the right practices? Is it occurring during the phase of harvest? Is it occurring during the phase of transport at the final center? And then also during preparation and even post-cooking. So all of these phases can essentially be associated with contamination that can occur. Also, the pathogen is important. So if you compare Salmonella versus Shigella, you all must be very well aware that Shigella requires a much lesser dose of the organism in comparison to Salmonella, that requires a much higher number of organisms. So the proliferation rate of the organism also matters. And then survival of the pathogen in food. So if you see organisms like Listeria, they can survive even in smoked foods or in refrigerated foods rather than smoked. You will see that these organisms can survive different temperatures, which is why it is important for us to understand each outbreak in totality. So the key concepts as per CDC is a sick food worker contaminates ready-to-eat food through bare hand contact. A sick food worker contaminates ready-to-eat food other than through contact through utensils which are contaminated, or a sick food worker may contaminate food through even glove hand contact. So you may see multiple times in your own hospitals or in many institutions that there is a single glove kept that the person who's serving the food actually keeps wearing and removing the gloves to be able to pick up and serve food. So through glove contact also, infections can spread. And food handling practices, the temperature at which they're stored. We all know that organisms like Clostridium can exist even if you cook them and then refrigerated and recooked. If they're spore forms, they can actually become active forms and be a source of infection. So the causative organisms, as far as foodborne illnesses and diseases, are classified into three. You have foodborne infections, you have foodborne intoxications, like, for example, Clostridium botulinum or Staph aureus, and then you have toxin-mediated infections like enterotoxins that can be caused, like, say, for example, the Shiga toxin or the Shiga toxin-producing E. coli also, which are very, very important. So I'm coming to a list of organisms that have been associated with foodborne illnesses and have been associated with outbreaks in various times. I'll focus on each of these individually. Only the major outbreaks I will talk about, but just to give you an idea of what you need to consider when you have a patient with a foodborne illness, or rather patients with foodborne illness. So Bacillus cereus is associated with food poisoning, with meats, stews, and gravies. This, in fact, we also call it the Chinese fried rice, kind of diarrhea that causes abdominal cramping and watery diarrhea and nausea. I don't know, many of you from Mangalore will also know that there was one of the Chinese places that we used to really, frequent at that point of time. And essentially every one of us used to get diarrhea post, visiting that place. And they shut it down now. But essentially, we don't know what the causative organism was, but it would be interesting to understand what those organisms were causing that kind of diarrhea. You had Campylobacter jejuni that is associated with raw or undercooked poultry. So even the salads that you eat, which are made of chicken, for example, or unpasteurized milk, can cause Campylobacter jejuni diarrhea. It can cause diarrhea. Bloody diarrhea also is very common. Cramps, fever, and vomiting. Next is Clostridium botulinum, which is, of course, we are always associating it with honey, but you have other canned foods also. As we are getting more and more, produce that is canned, that could be another source. Tuna, canned tuna, for example. Clostridium perfringens also present in meat, poultry, gravy, and dried foods can be a source of infection or outbreaks. Shigella is quite a common source, again, from raw produce or undercooked food. So many of the time it's the lettuce which is undercooked, which is there in our burgers, that is a source for outbreaks from Shigella. Staph aureus can occur even in unrefrigerated meats or cooked foods where you cannot isolate the organism, but the toxin is still present. So it can be in egg salads and ice creams. Okay, so that is again something very, very important. Next comes Vibrio parahemolyticus, again undercooked raw seafood and shellfish. Vibrio vulnificus, again seafood and shellfish. E. coli 0157H7 that was involved. Of course, many of you may have read the book from Robert Cook also on this, where they were undercooked hamburgers that were the source of infection. And then you had Listeria monocytogenes also causing outbreaks that are there. So foodborne illness investigation has to look at environmental health assessment, foodborne illness outbreak, epidemiological assessment, and laboratory analysis. So how do you actually look for it in the community? Because many of these are community outbreaks. Prepare for a field investigation, report it as a foodborne illness, because many times unless the authorities are aware of it, they are not going to actually look into it. So it's important for them to be informed about it. You have to verify and confirm the diagnosis. You have to construct a case definition and identify cases, perform descriptive epidemiology while line listing each of these cases. You have to develop a hypothesis of what is commonly consumed by all of these places. Was it where, every one of these people attended say a marriage, dinner, or they had been to a particular restaurant, or they were part of a particular cruise group. So all of these could be possible. Then you have to test the hypothesis by looking at the organism. All right, and then conduct an environment health assessment. The HACCP checklists are there from the FSSAI, so they can be looked into also. Implement control and preventive measures and report communicable findings. So all of this has to be done. Then you have to prepare an epi curve. Now, again, this has been dealt with the other talks in brief, but I'm also going to add on to that. So once to be able to look at an outbreak in totality, here you see different curves. So what does it tell you? If you look at curve number A, you will see that it's a point source epidemic that started off and then built the number of cases, and then you had the number of cases decreasing over time. Whereas if you see the second one, it's a continuously source outbreak where you have high numbers all along the outbreak. Then you have, the third case, which is an intermittent common source outbreak, where there are spaces of time involved during the outbreak period where there are no, there is no, cases isolated as well. And then you have, a propagated outbreak where there is a person-to-person spread, where continuously you will see that cases are being recorded at least at a basal level of a minimal amount. So let's look at the major outbreaks according to the CDC that have happened in the US. So in fully cooked chicken in 2021, listeriosis has been, isolated. Also in the queso fresco, listeriosis has been isolated. 2020 had deli meats looking at listeriosis. You also had enoki mushrooms being a source of listeriosis. 2019 hard-boiled eggs had listeriosis. Listeria monocytogenes infections again in the daily sliced meat. Pork products again listeria. So you see the last 3 years has been all about listeria in the US outbreaks. I was quite interested because the other day when I was talking to my niece who lives in the US, she was telling me that her mother does not allow her to have poached eggs with runny yolks because they are worried of salmonellosis, and they're not medicos. So it was interesting to see that they felt that this is important. Similarly, 2017 and 16, again, you have a lot of listeriosis that has been a cause of outbreaks in the United States. Now I'm coming to a very important outbreak. So I'm only going to be dwelling with a few of them, which really impacted history in recent years. So in 2011 of May, there was a huge outbreak in Germany, which is such an advanced country with such good state-of-the-art technology, where they found that hemolytic uremic syndrome was discovered in more than 3,816 patients, which included 54 deaths. 22% of which had hemolytic uremic syndrome. And they actually looked at this organism, E. coli O104H4, which is an ESBL producer. But they thought the source of this outbreak was from Spain, the cucumbers which had been bought from Spain. So they actually discarded scores and scores of cucumbers, thinking that this is a possible source. And finally, they kept really looking for the source. So they looked at the seed population, the diversity in French outbreaks, and then in German outbreaks. And then they looked at various other sources as well. They did a variable mutation for a rate hypothesis, a bottleneck hypothesis. So all these hypotheses were put forward. And finally, they were able to trace the source of the organism to sprouts that had been imported from Egypt. So a trade network for contaminated sprouts led from a producer A in Lower Saxony to 26 sprout distributors, which are the black dots that you see there. And then from there, 41 identified outbreak clusters, to which they were able to then see how the whole spread occurred. So this is the epi curve of the different disease outbreaks that was there in terms of the sugar toxin like gastroenteritis and the hemolytic uremic syndrome like gastroenteritis in males and females. And you will see a clear spike in the number of cases during May, which really got them very, very nervous about that. I think all of us remember with outbreaks and salmonella, we cannot forget remembering Typhoid Mary. So Typhoid Mary, Mary Mellon was a cook who used to work in the rich houses, and she and restaurants that were in New York, and she kept moving her jobs because people whom she served food, especially the householders, got sick. And she, if you look at a beautiful series on, I think this is on Hotstar, on Mary Mellon, you will see that she used to make ice creams that were really phenomenal, and they were the source of salmonella. She was implicated initially by the public health officials who actually put her in an isolated island, but they could not prove her because organisms were not isolated at that point of time, remember, so conveniently, and never had they been able to isolate an outbreak like that to a single point source. So she was let back from there, and she contaminated and spread infection with many more, because nobody was able to prove that she was a chronic carrier of salmonella in her gut. And she was the source of infection to many, many. So there have been a number of salmonella outbreaks in the US that have been associated with peanut butter, with the ground turkey, with Foster Farm chickens, with Mexican cucumbers, and that of E. coli from hamburgers, Dole baby spinaches, Taco Bell fast food, also in Chipotle, and in the hot sauce and canned potatoes, botulinum toxin outbreaks have also been reported. Listeria outbreaks include the cheese, the hot dogs, the turkey meat, and the cantaloupes that were essentially then discarded. So there was a farm to fork investigation of an outbreak of Shiga toxigenic E. coli 0157 that was conducted. And here they were able to link it from a cross-contamination of the raw meat to the foods at the butcher's premise, which was then the most possible route of transmission that they highlighted. And then they found that they had to take measures, or risk assessment was conducted, and mitigation measures had to be put in place to reduce that. So this is the epi curve for that particular outbreak of the 0157H7 that occurred in the previous paper that I shared with you. And you will see across a time period, there were cases that were reported over very short time periods of about a month. I'm now coming to another community outbreak of Salmonella enterica and the use of whole genomic sequencing for identifying the source. So it's very important that just isolating the same organism, because as you know, one Salmonella is a huge organism profile. So unless you do serotyping to identify each subgroup, that particular group, in fact, even the susceptibility profiles of Salmonella, as you know, are given as a whole, whether it's CLSI or UCAS. They don't give Salmonella enterica, typhi as one group, or for the C. They only look at typhoidal and non-typhoidal Salmonella, which can cause infections in bloodstream as well as gastrointestinal infections. So this also, they found that there was human epidemiology confirmed there was an unknown epidemiological link from the outbreak 5. There was human unlinked epidemiologically, what you see in yellow in outbreak 2, and then food being the source essentially everywhere, whether it is outbreak 1, 2, 3, 4, or 5. So outbreak investigation is actually very interesting for us to do. Foodborne outbreaks due to bacteria in Taiwan from 86 to 95 also looks at organisms like Vibrio parahemolyticus, Staph aureus, Bacillus cereus, E. coli, Salmonella, Clostridium botulinum, or others. So how do you really go about it? Because organisms are so difficult to really speciate and figure out. So basically, bacterial outbreaks, it's best to do next generation sequencing, genome assembly, annotation, and comparative genomics. You can also look for pathogenicity and antimicrobial resistance. Again, the antimicrobial resistance and pathogenicity may be plasmid-borne or prophage-borne, or maybe a genomic island, or maybe associated with SNPs also. And these can be either strain-specific typing methods may be created, or treatment decisions also can be, helped with when you have this kind of data. So it's very important that you use the current, modules that are available for outbreak diagnosis. And I understand that many a times it is difficult for us to do, especially in a country like ours where we have to outsource, tests like sequencing. And but it's important to store your strains adequately well, especially when you have an outbreak. So here you will see again the sequencing to track bacterial infection outbreaks, and you'll see whether it is Klebsiella pneumoniae or Staph aureus. Genomic sequencing or NGS is a very good tool, or MLST is a very good tool. So here you'll see the CDC again had put out an investigation and control of vancomycin resistant Staph aureus in an outbreak that was there. Now I'm coming to another outbreak that was there with sugar toxin producing E. coli of the serotype O117H7 among men who have sex with men. So this may not essentially be only a foodborne outbreak. It may also be a sexually transmitted outbreak. You have even Neisseria outbreaks that come out like that. So here you will see it was typed across Asia, Africa, Latin America, Pacific, and no travel across years 2006 to 2017, and antimicrobial resistance like azithromycin resistance was also checked. Male and female were also tested. The other important source of outbreaks is water, because that's something we all consume. And as years go by, if we look at the WHO predictions, within another 10 to 15 years, we are going to be out of fresh water in the world. Good fresh water is going to be an asset that we will be paying millions for. So considering that, the biggest organism that we have to look at is the Vibrio cholerae outbreaks that have occurred across time. There have been 7 huge outbreaks that have swept across the world. And if you look at that, it's the first pandemic, second, third, fourth, fifth, sixth, and you see the 7th pandemic. Actually, you can see the movement of the, the way the cases spread across the world. So if you look at this, we have among the Vibrios that we are really interested is Vibrio cholerae, Vibrio parahemolyticus, and Vibrio vulnificus that are associated with mollusks. Many a times I wonder why we don't really isolate so much of Vibrio parahemolyticus or Vibrio vulnificus. It may be because we cook our food well, but it does not mean that it's not there as a surveillance tool. And I want to bring your attention back to Haiti, where the outbreak of Vibrio cholerae occurred there after the big earthquake. And it was, finally traced back to the Nepali helpers who had come down to help the, people who were suffering there. And they were the source of the outbreak. And from there, it spread across, causing so many deaths also. So next, coming to Vibrio parahemolyticus and the pandemic potential. So suddenly in 1996, there was a flurry of cases that were of the serotype O3K6 that were reported from Calcutta, Nitte Institute. And from there, also other cases that were, in the whole of Asia that were reported in 1996. But 1997, this same variety of O3K6 was also reported in Peru along more than 1,500 kilometers of the coast to reach the Chilean border. And then several thousands of cases were reported in 2003 and 2004, with 10,000 people being affected in Chile. Now, when they actually tracked this, so they found that the global spread was associated, like you see here, you have it in India and many Asian countries. And then you see it across this border of the town, of the countries in South America. And this was actually associated with the tides, the El Nino effect, where the warm waters, as they spread, took the organism along with them, and the same strain was seen across there. So the molecular studies were able to prove this, that the 1997 El Nino episode provided the actual, the platform for the spread of the organisms from the coast of India and Asia across to the South America. Just give me 1 minute. Sister, I'm not able to listen. Can you please keep quiet? The El Nino phenomena is characterized by the arrival of equatorial warm waters in a sequence of waves, and 6 months it took for more than 10,000 people to be affected. It has been suggested that a recurrent invasion of the tropical masses have resulted in this kind of this thing. So this is how the spread of the emergence with the El Nino effect that has been recorded in various papers. So you will see here how the spread of this has also been associated with Vibrio cholerae, that has had similar spread associated with climatic changes that have occurred. Now, many of us are associated with hospital-based outbreaks. We have Klebsiella, multi-drug resistant Klebsiella outbreaks that are there, or Pseudomonas outbreaks that are there, multi-drug resistant Pseudomonas, and many others. So I'm bringing your attention to a couple of outbreaks that that can actually occur in a hospital. You can have community outbreak of conjunctivitis that was in Minnesota, that was reported with Streptococcus pneumonia. Again, you see an epi curve here with clinical conjunctivitis that was there in so many cases, but culture proven only in few, and you have to record all of these things. And we cannot complete a hospital outbreak discussion without talking about Clostridioides difficile. It's no longer Clostridium difficile. So I'm, please note that change. But this paper was way back in 2012 when the name was still Clostridium difficile. So Clostridioides difficile again has been associated with various outbreaks. It's being a very hardy organism that cannot be cleaned with alcohol or hand rubs with alcohol. It requires soap and water. The alkaline soap is a much better source of, infection prevention as far as stopping the spread of Clostridioides difficile. You will see here that there is an onset in 2002, which went on to 2003 and 2004. You have multiple outbreaks that have been associated with long-term care facilities, especially because of the source being so difficult to eradicate. This is our whole genome-based sequencing of multi-clone Serratia marcescens. Generally, Serratia is associated with neonatal ICUs in tertiary care hospitals. But this particular outbreak, we actually looked at a series of cases that came over a particular short about 6 months of time. So we had, once we did the NGS, we figured out two big clusters that was there. One was the cluster that was associated with the CCU, that is the cardiac coronary unit. And then this was the other major outbreak in yellow that you see from the adult ICU, sorry, the one in peach. The yellow was the extensive surveillance. So it took us 6 months to actually figure out the cause, and we were really looking at every place possible for surveillance of these organisms, and we were able to identify the source of the organism. This is our epi curve, and you will see here there is a typical time period in between where no cases were involved, and we were not able to figure it out, but we were finally able to get it from the environment. That is, we were talking about the Hepsol solution that was used, which was reconstituted, or we had it from the suction bowels of the patient. These were the sources in the environment that we had the organism from. But this was not a multi-drug resistant organism. It just was a increased number of bloodstream infections that we have had with this organism. Next is the AMR based outbreak, and that I think is an important topic today when we are in a COVID-19 pandemic. We have forgotten that we are also in an antimicrobial resistance pandemic that is going on at such a slow pace that many of us don't even realize that we are in a pandemic. So, even accepting that this could be there is, a huge problem. So here I'm describing an enterohemorrhagic E. coli O121H19 that had an ESBL gene that caused an outbreak across 2 nurseries. And again, whole genome sequencing helped to identify the source of the outbreak. You see here, you have nursery A in blue and nursery B in green. And you have the spread of the infection across time of 1 month across the nursery A to nursery B, where also there was some joint childcare that was associated with family members and asymptomatic carriers being possible sources for the spread. So they basically found that health centers should take measures to prevent the spread not only of primary infections but also secondary infections and antimicrobial resistance. And it's important that extensive monitoring and control should be in place as far as AMR is concerned. A series of the next few slides are going to be from the ECDC's time distribution of hypervirulent Klebsiella across different countries in the EU and in the rest of the world. And you will see that they are looking at certain strains being more common and the outbreaks being across time. Here is again the hypervirulent Klebsiella ST23 that has, you will see that KPC2 is quite common. And if you see a long time, as you get closer to 2019, 2020, this landscape is shifting from KPC2, which is more common, to OXA48 and OXA232, which is seeming to be more common as an organism. Next is again, this is again hypervirulent Klebsiella ST23, that is sequence type 23, where you have Vim and OXA48. Again, Vim being only in 2016, but OXA48 becoming a more common, source of organism. And within the EU, you will see here that they have looked at about 348, different isolates from different countries. And you will see here the rate of isolation is gradually increasing. 2020, I would think that it is a little less because of the COVID outbreak, where the admissions itself dropped down from normal causes that people underwent, say surgeries, for example, planned procedures were all cut off and only COVID care was there. So there was a change there. And you have a number of OXA244 producing E. coli ST38 of cluster A and cluster B across UK in 314 strains. And again, you see cluster A is the dominant strain across the recent years. Next, just to finish off, I have a couple of respiratory outbreaks that are there. You have Corynebacterium coming up again, diphtheria and non-diphtheria. Pseudo-diphtheriticum here was a huge outbreak that was there in Myanmar nationals, displaced Myanmar nationals in 2017 to 2019. Then you have bioterrorism being associated with outbreaks. And here I want to bring your attention to anthrax, which was used in after the 9/11, where public health importance of knowing about bioterrorism became so important. Before I just finish off, because there's no separate topic on algal toxins, I want to bring your attention to that. Just the last couple of slides. Here you see harmful algal blooms or the red tides that have been causes of various other infections and outbreaks, like the paralytic shellfish poisoning, you have also the outbreaks due to cyanobacterium, which is, of course, bacteria is not algal, but that can be another bigger source. Storms, hurricanes, increased nutrient inflow because of heavy rains, the surface temperature changes due to which fish kills, shellfish toxicities, Ciguatera fish poisoning also is important. So here, you have the paralytic shellfish poisoning in Mangalore in the west coast of India that was reported near Kumble, where there were quite a few oysters, and people who ate oysters were all complaining of tingling and nervous symptoms that were associated. A lot of them were hospitalized. There's another outbreak of paralytic shellfish poisoning where the toxin profile was created. You also have other outbreaks that have been talked about. And then comes the Ciguatera toxin that is associated with eating the head of fish that is there. Again, got 100 people sick in Mangalore. So to complete today, just to take home, travel restrictions during a pandemic. So I think all of us have, made it through 1.5 years, not realizing that we could actually complete a lot of our work without traveling at all. So this particular CME, for example, would have been live had it been, and I cannot, I can in no way say that me not being able to see the audience is any better, but travel restrictions during a pandemic are very, very important. Had it been put in place, in fact, in Wuhan much earlier, let's forget about the fact whether it was a hospital, lab outbreak or was it a natural infection, but restriction of travel would have probably helped curtail the pandemic much earlier than spread it to proportions that we are actually seeing right now. Land use and urbanization, like Dr. Ravi spoke on his talk, many a times we are encroaching into areas that are not suitable for us, and that is when we are susceptible to outbreaks. Like I told you about the El Nino effect, climate change is real. Let's come to think that we have to understand how to live with nature, not let nature live like the way we would like it to. And increased contact between man and animals. A lot of zoonosis happens nowadays, which has not been in the past. Vector control, because a lot of outbreaks are associated with vectors. Global surveillance. So we are, as India, we are not part of the One Health campaign, where veterinary, agriculture, and human health has come together under one umbrella. But yes, I think it's important that we start thinking towards those lines: One Health and antimicrobial resistance. Use of NGS and WHGS, that is next generation sequencing and whole genome sequencing, is very important for outbreak analysis. Development of point of care diagnostics. Now, why is it's so easy today? I don't think ever in history have we been able to develop diagnostic kits within a span of 1 or 2 months from the start of the pandemic. So even we started testing way back in April 2020 for COVID-19, and we've completed over 50,000 tests now. Point of care diagnostics is what we use. So we use the TrueNat type of system where you are able to give a generator report, a confirmed report within 1 hour of diagnosis. So that is very, very important that we focus towards point of care diagnostics and diagnostics as a whole. And vaccine development, again, a very important thing today. Yesterday, we have touched 1 crore vaccine in a single day in India. And that's a huge, essential take home, that vaccines, we need to keep looking for vaccines. Today, if the mRNA vaccine has come out so early, it is because of years of research that took place before it. Or for example, the adenoviral vector-based vaccine like AstraZeneca, again, this was years of research on using Ebola models, for example. So to complete, I think it's important for us to realize that, microbes are a huge source of outbreaks, especially bacterial, bacteria, and we cannot forget antimicrobial resistance. Thank you so much for your kind attention.
Thank you so much, ma'am, for that beautiful talk on bacterial outbreaks. There is so much interesting history about these outbreaks, and thank you for sharing a few important ones with us. We have a few questions if you would like to take them. Internationally, antimicrobial resistance is a huge thing. So even if they find 1 case of AMR bug, they will work it up, they'll follow it up, involve the CDC. Is the same thing happening in India, or is the trend at least switching towards that side?
Okay, so this is a huge question that you touched upon, and I'm glad you actually brought it up. So when I went for an observership in the University Hospital of Basel, Switzerland, every Monday there was MLST done for every isolate of MRSA that was isolated over the week, and there was a discussion between the microbiologists, the ID team, and the clinical departments that were there. And they were discussing the weeks that I was there, the number of isolates were only 2 or 3, that they had such extensive discussions over an hour on discussing where this could have spread from. So, I actually talked to our professor there, my professor, and I asked him, why are you really looking at this? Because it's just 2 cases. It may not even be an outbreak. So why are you really, doing so much of, a surveillance for this? And he told me, Anusha, it's important that you, not allow antimicrobial resistance bugs to actually snowball into an avalanche when you cannot manage it. And that actually taught me a huge lesson. So we are talking about ESBL rates of 80%, 90%, carbapenemase rates of about 20% or 30% in many institutions. And it's because of spurious use of antibiotics, low quality antibiotics, over-the-counter medications, and lack of evidence-based practices that are being followed. So in considering that, I think we have to take a cue from that and understand that it is important that, there are so many students now. Medical colleges have students who would like to do small projects. So it's important for them to understand how to do a line listing, how to do an outbreak investigation. So each of these can be given as a topic for them to study, and that will help them understand the impact of it, because they'd be reading more of it, and eventually we will be having more data, because right now we have no data from India. Most of what I presented was from abroad. So it's important that we have data, and for which I would really encourage everybody to get more vigilant.
Yes, ma'am. And I think COVID has boosted the pharmacy industry so much that there are a lot of antivirals coming up. Are there any new antibiotics coming up? Because right now, many AMR, we have nothing to treat the patient. It is just wait and watch whether they survive or not.
The antimicrobial, the development of new antibiotics, actually the pipeline is really running dry. And the reason for this is completely, I think the pharma has to take a larger role, because it's easier for a pharma to prepare an antihypertensive or an anti-diabetic that a patient is going to take for the rest of his life than a 5-day course of an antibiotic. In fact, if you see many of the medical representatives who come to talk to you, they will be encouraging you to use more antibiotics, because essentially they'll say, why don't you add clindamycin into your repertoire of antibiotics that you're already going in for the patient. So I think pharma has to understand. We have a few newer combinations that have come up. Ceftaroline, you have ceftazidime, avibactam has been there for a while now. We have meropenem, viborbactam. We have quite a few, but we are especially for gram negatives, and you have for the gram positives, you still have a few more delbavancin, you have a few of them, but I think we are really hitting rock bottom as far as antimicrobials are concerned. It is important that we use what we have judiciously, rather than overuse, try to prophylactically use, empirically use with no evidence-based medicine and only eminence-based medicine. These are something that are very, very important.
Yes, ma'am. And you think clinical features like fever with chills, does it help us diagnose whether we are dealing with a bacterial or viral infection, or could it be a presentation of both of them?
Right. So again, of course, being from Kshema, I've had very strong roots because of the, the kind of clinical acumen that was inculcated in all of us. Even today, to this day, we go on rounds. We actually look at patients ourselves, though we are in a lab field. But what we realize out of the whole story is that there is a big problem with, not being able to diagnose something and starting on a prophylactic antibiotic or an empirical choice of antibiotic without knowing whether it's bacterial or viral. So it's important to send the right investigation and the right sample to the microbiology lab. Talk to your microbiologist, because with microbiology, it's garbage in or garbage out. So if you give a sample, like you've taken 5 liters out of a ascitic fluid and send 1 ml to a microbiology lab, your chances of recovery are lesser. So important to diagnose the source of infections. For example, in the question that you asked me, fever with chills, could be viral, it could be malaria, it could be bacterial infections, it could be a urosepsis with a urinary tract infection. So unless you have, say, a routine urine culture, or you have blood cultures, or you have now dengue, malaria, etc., so easy to diagnose. It's no longer like when the old GP used to practice with no diagnostic help at all. Use diagnostics to your support. You don't, I would really suggest everyone to not feel insecure about, talking to a microbiologist or talking to a lab to be able to use a diagnostic test. The ultimate goal is patient care, and better patient care comes with multidisciplinary approach where all disciplines actually contribute towards patient care.
Thank you so much, ma'am, for so patiently answering all our queries. And thank you for taking time from your busy schedule and being with us today for this presentation. Over to you, Siddharth.
Thank you, ma'am. Thank you for the lucid presentation. If we do not know where we come from, how will we ever know where we are going? Today's lecture series gave us a bird's eye view of what the history was, what the present is, and what the future possibly holds. And like all good things come to an end, we've come to the end of our lecture series. I now request Dr. Shoaib Ahmed, Assistant Professor in the Department of Internal Medicine, SHEMA, to deliver the vote of thanks. Over to Dr. Shoaib.
We have one postgraduate, final year postgraduate, Dr. Srivatsa. He will be presenting his paper, following which we'll have the vote of thanks. Siddharth?
Yes.
Okay.
Dr. Srivatsa has been added. He's a co-host with us. He's going to present to us a research paper that he has written and presented yesterday, which has also won the second prize. I'll shortly read out the presentation topic. The presentation was neutrophil lymphocyte ratio and platelet lymphocyte ratio as a predictor of mortality in severe COVID-19 cases. He's won the second position amongst 7 in the competitive category. I now invite Dr. Srivatsa to present his paper. Over to Dr. Srivatsa.
Can you hear me, sir?
Yes, you're audible, Srivatsa.
Good evening. Good afternoon. So I'll be presenting a paper that is neutrophil lymphocyte ratio and platelet lymphocyte ratio, which is used as a correlator of mortality in severe COVID-19 cases. With this research was done with the help of Dr. Ellen Samaga, sir, Professor and Unit Head, and Dr. Supriya PS, Associate Professor. So as we know that COVID-19 illness caused by SARS-CoV-2 virus is a highly infectious disease, which is rapidly progressive and sometimes fatal. The severe inflammatory response syndrome has contributed to a weak adaptive response, thereby leading to immune response imbalance, due to which many of these patients have rapidly progressed from mild to severe cases. Circulating biomarkers, which represent inflammation and immune status, can be used as potential predictors for the prognosis of COVID-19 patients. So in this study, we are aiming to do so by using NLR and PLR as biomarkers to predict the mortality in severe COVID-19 patients. We know that there are already numerous inflammatory markers that have been studied, most of them being expensive, and there is a significant time delay which may jeopardize the patient treatment. Similar studies have been done in the past, and majority have been reported from China and Europe, with very few studies done in our country. So hence, this is a retrospective study, a cross-sectional and observational study conducted in our hospital, which included 224 adults aged above 18 years and who were diagnosed with COVID-19 illness, either via RT-PCR or CBNAT. Blood was drawn from them, and various ratios and blood parameters were calculated, which included total leukocyte count, from which the absolute neutrophil and total lymphocyte counts were calculated, and a ratio NLR ratio was given. Similarly, using the platelet count and total lymphocyte ratio, total lymphocyte count, we calculated the total PLR ratio, the CRP, D-dimer, and ferritin was also calculated. The patients were then characterized into mild, moderate, and severe categories based on the guidelines given by Ministry of Health and Family Welfare India. These results were then analyzed via descriptive statistics. So based on the results that we obtained in our study, we found out that majority of our patients were male. That is 151 patients of the 224 patients were male, and 73 of them were females. That accounted to 67% male and 33% females. When considering their age, the mean age among the males were 45.69 years, and among the females were 51.6 years. There was significant NLR and PLR ratio differences between the age and gender, because we know that NLR and PLR ratios can be varied with the gender and the age. The mean NLR ratio among the women were higher, that is about 9.27, when compared to that of men, which is 6.53. Similarly, the PLR ratios were also found to be higher among the women when compared to men. The mean NLR ratios amongst patients less than 60 years of age was 8.96 when compared to those with patients among greater than 60 years of age, which was higher, which was 9.83. This probably could be because of associated comorbidities associated with elderly. There could also be undetected malignancies or inflammatory conditions, occult inflammatory conditions, which might be going on in them, that is patients above 60 years of age, which go undetected. Hence, probably the NLR ratio is higher among the males. Similar studies were done elsewhere, which also showed that the NLR ratios varied with age and was higher amongst elderly when compared to the younger patients. Among the differences in women and men, there were no studies which showed any difference between men and women in terms of NLR and PLR ratios. However, in our study, we found that the females had a higher NLR ratio when compared to that of the males. We've also tabulated the inflammatory markers that we've calculated in these patients, and we found out that there was a positive correlation between mild and moderate cases. We found out that the NLR ratios, the average NLR ratios in moderate to severe cases was 14, when that in mild was just 3.6, with a statistical significance of less than 0.01. Further, through area under the curve and statistical analysis, we also found out that the NLR ratio corresponding to more than 6 was statistically significant and could be used as an independent prognosticative marker for various diseases. We then used these NLR and PLR ratios and to calculate, we made a 2x2 column to calculate the sensitivity and specificity to know the severity of illness and the mortality of COVID illness due to COVID illness. So we saw that patients who had an NLR ratio more than 6 had a specificity of 87.6% and a negative predictive value of 86.18% in predicting the severity of COVID illness. We also found that when the NLR ratio was more than 10, it has a specificity of 94.21% and a positive predictive value of 89.86% in predicting the mortality due to COVID at the time of admission. PLR ratios were also found to be important, with PLR ratio more than 300 having a specificity of 92.56% in predicting the mortality due to COVID at the time of admission. So to discuss the findings, we know that the main pathophysiology of COVID involves cytokine storm and endothelial injury. So we needed markers which indicate inflammation and immune status. Amongst the WBCs, we have the neutrophils and the lymphocytes. The neutrophils mainly are markers of inflammation and the lymphocytes are markers of immune status. So using these, we try to establish a relationship between the mortality and the severity of illness amongst these COVID patients. So our data showed that the NLR ratio at the time of admission is high, showed high mortality, corresponding to high mortality. Follow-up NLR and PLR ratios could have been done. However, they may be altered because of the use of steroids, antibiotics, and other anti-interleukin treatments, which will modify the NLR and PLR ratios. When patients above 60 years had an NLR ratio with an NLR ratio of more than 10, we saw that 53.2% of these COVID patients immediately progressed from mild to moderate disease to severe disease, and all of them succumbed to death. The NLR ratio had the highest area under the curve when compared to ESR, CRP, and ferritin at similar values with a high sensitivity and specificity. It also showed that the NLR ratio had a high diagnostic value to predict mortality when taken at the time of admission. Similar similar studies and meta-analysis were done in various other places, mostly in China and Europe, which also showed that the NLR had a pooled sensitivity of 78% and a specificity of 78.2%. However, in our study, we found out that the NLR ratio to predict mortality had a sensitivity of 83.5% and a specificity as high of 87%. So to conclude, that we have the NLR ratio, which has a very good predictive value on the disease severity and mortality in COVID-19 cases. Why NLR ratio when compared to the other inflammatory markers is that it can be used in resource poor settings, as it is easily available, it is very cheap, easily measurable and reliable, and can be interpreted by any person. It can also be used as an early warning sign when to refer the patient to a higher center from a primary health care center. It can also be used as a useful parameter to triage the patients into mild and moderate to severe cases when the patient arrives in our casualty. We can also use it to optimize treatment with steroids, anticoagulants, antivirals, with early identification of severe COVID-19 illnesses. However, NLR ratio had a number of limitations because, as we know, NLR ratio is associated with immune inflammatory response, neurovegetative states, and endocrine stress. NLR ratios can get altered when there is organ dysfunction with increase in steroids, increase in cortisols, etc. It can also get altered with the presence of intercurrent illnesses like bacterial, viral, and fungal illnesses. Previous studies have been shown to use NLR ratios for malignancies and pharmacotherapies. Inflammatory disorders also tend to elevate NLR more than the non-inflammatory disorders. So by this, we can use NLR ratios as a more beneficial tool than the other inflammatory markers. Thank you.
Thank you once again, Dr. Srivatsa, and congratulations on your victory. I once again invite Dr. Shoaib Ahmed, Assistant Professor in the Department of Internal Medicine, to deliver the vote of thanks. Over to you, sir.
Thank you, Siddharth. Mute the volume. Okay. A very warm good afternoon to one and all. I'm Dr. Shoaib, Assistant Professor from the Department of Medicine. I would like to start by saying it's a great honor and privilege to render the vote of thanks for today's program. On behalf of Department of Internal Medicine, SHEMA, first and foremost, I would like to give my heartfelt gratitude to our Honorable Vice Chancellor, Dr. Satish Kumar Bhandari, for giving the inaugural address and constantly pushing us for organizing such CMEs. I would also like to thank our beloved Dean, Dr. P.S. Prakash, for your opening statements, opening remarks, sir, and active encouragement. I would like to thank all the distinguished and esteemed speakers, Dr. Yamuna Devi, Madam, Dr. Ravi, sir, Dr. Nitin, and Dr. Anusha Rohit, ma'am. First of all, for readily accepting to be part of our program by taking time off your busy schedule, and also for your brainstorming and enlightening sessions on each of your topics for us to be better prepared for the next outbreaks. Thank you, speakers, for your knowledge and time. I would like to thank the President of API DK Chapter, Dr. K.S.HEGDE, for co-organizing this event and supporting us. I would like to acknowledge the judges for the paper and poster presentation, which was held yesterday, Dr. Shyam Prakash, Dr. Tanmay Bhatt, Dr. Asha Pai, and Dr. Nikhil D'Souza. Thank you, sirs. I would like to thank our students, Kripa and Megha, for gracing the occasion with a prayer song. I would like to thank all the authorities of Nitte, deemed to be university, the IT department headed by Mr. Mahesh Chauta and his team, the Deputy Director Sai Prasanna Hegde, the Faculty Development Cell Director Mrs. Sadhna Deshmukh and a team, as well as Bharti, who has helped us with the technical assistance, and also everyone behind the scenes who has helped us in making the CME a successful one. No CME is complete without its delegates and participants. So I would like to thank all the delegates and participants for your active participation in making the CME a successful one, and hope it has helped you. And last but not the least, I would like to thank our HOD, Dr. Sudhindra Rao sir, for coming up with this idea, and all my colleagues in the Department of Internal Medicine and the postgraduates and interns who have been running around for the last 2 weeks and getting things done to make this program a grand success. This is the first time we have organized a hybrid CME, and I'm pretty sure this is first of many more to come. Thank you. With this, over to you, Siddharth.
Thank you, sir. With this, we come to the end of the series part of our CME. The last leg of the hybrid CME is a quiz for postgraduate students, and we've confirmed participation of 6 teams from 6 different colleges of our state. We'll be starting with the quiz at 2 p.m. and it'll be taken over by Dr. Rohit Rajivan, Assistant Professor in the Department of Internal Medicine, and Dr. Harshita K. Punja, who's a Senior Resident in our department. I thank you all once again. I thank all the organizers who were involved with the meeting off and on camera, and all the delegates who participated and benefited from our program. Thank you all. See you all at 2 p.m. when we begin with the quiz. Thank you once again.
Yeah. Very good afternoon to all of you. We are we had a great academic feast today morning with a lot of renowned speakers and who have provided a lot of knowledge and insight for us to in our day-to-day practice with regards to all infections and how to manage them. And now at the end of this, we would like to finish off with a bang, and hence we have a quiz. Okay. And I hope all of you are ready. So we have we have 6 teams here joining us today. We have AJ, we have Yanapoya, KMC Mangalore and Manipal, Sulya and Shema, not last but not the least. Okay, so can you just give me the team numbers? I'll announce the team numbers once again for everyone's convenience. So team 1 is the team from KVG Sulya. Team 2 is the team from Yanapoya. Team 3, KMC Manipal. Team 4, KMC Mangalore. Team 5 is the team from Shema. And team 6 is AJ Medical College. Okay, I would like to request all of you to keep your cameras on, whoever is participating in the quiz, please. Okay, yeah. So I will now share the rules. Okay. Okay, you are able to see the slides, I hope. Yes. I know. No, it is hanged. I selected the slide. It's hanged. Everything is. Just hold on, please. We're having some technical difficulties.
It's actually muted.
Oh, yeah, are the slides visible now?
Yes.
Yes, sir.
Yeah. Yes. Yeah, thank you. Okay, so the quiz is for 1 hour. There will be 4 rounds, and yeah, the audience is advised to keep the mic muted. We have 4 rounds. With round 1 is pandemic moments in history. Round number 2 is categories. Round number 3 is connect, and last but not the least, we have a rapid fire round. So I'll give you a run through all the rules of each round. Okay. So round number 1, we have 12 questions with no negative marks. Okay. Each team will get 2 questions. Okay. You have a direct question where you get 20 points. You get 40 seconds to answer that question. A pass question. So if you don't know the answer, you can pass it. Or if you don't answer within 40 seconds, the question goes to the next team. For example, if team number 1 gets a question, they do not answer in 40 seconds, the question gets passed over to team number 2. and team number 2 will get 15 seconds to answer that question. If they don't answer in 15 seconds, the question gets passed over to team number 3 and so on. All right. Round number 2 is 6 categories. Okay, you cannot pass them again, and there's 30 points for each correct answer. Okay. And round number 3 is where you get, it's called the connect round. In the connect round, we will show you images. The images will be shown in sequence. You will be getting 15 seconds for each image. If you are able to answer with the first image itself, you get 20 points. If you are able to answer with 2 images, you get 10. If you answer with the third image, you get 5. In the first slide, there will be 2 images. So you can connect the 2 and give an answer. In the second slide, there will be again 2 images. So if you get what's common among this thing, you can give the next. Okay. And again, there's no passing. There's no negative marks here too. Last round is a rapid fire round where you have each team getting 6 questions each. You get 45 seconds to answer all of them. The correct answer will get you 20 points. If you answer all 6 questions right, you get, in addition to the points, you get bonus of 30. Wrong answers will get you -5. Okay. So what you can do is you can pass the questions. Okay. I'll show you that thing. 1 second. Yeah. So you can pass the question. If you don't know the answer, you want to think about it. And if there's still time, you can go back to that question. Okay. In the rapid fire round. With categories, these are the categories available. Okay. So we have bacteriology, virology, mycology and parasites, vaccines, epidemiology, and last one, outbreaks in the news and in the movies. There's 1 question under each. Okay. So once that category is used up, then there's no way of getting it again. Okay. Yeah. So without further ado, we will start off with the first round, which is pandemics in history. And we will start with team number 1, which is the team from KVG. All right. 1 second, I'll load the slides. Okay. Yeah, KVG, I would like one of you to unmute the mic and just respond to me if you can hear me.
Yes, sir. We can.
Yeah. Okay. I would like, can you all introduce yourselves first, please?
Sir, myself, Dr. Amok, sir, third year junior resident, General Medicine Department, KVG Medical School. Myself, Dr. Vishwas, third year PG, General Medicine.
Yeah. Welcome to both of you. Okay. And and we'll start with the first question. Okay. Your time starts now. Okay. A trade deal with China unleashed one of the deadliest of these pandemics, and Italy was particularly hit hard. You have a map there for reference. So which among these diseases do you think is referred to here? You have plague of Justinian, the Ganges River cholera, bubonic plague, or COVID-19.
Bubonic plague, sir.
Okay. So that's the right answer. Okay, very good. Second team. Yeah. For Yanapoya, can you unmute your mics, please? Yeah. Can you introduce yourselves?
I'm Bharatesh. I'm third year medicine PG from Yanapoya.
I'm Dr. Sapna. I'm third year medicine PG from Yanapoya.
Yeah. Okay. Wish you all the best and welcome. So I'm giving you the second question. Yeah. Your time starts now. A London doctor used a dot map to pioneer the modern science of epidemiology. And these are the options. The third cholera pandemic, the Great Plague of London, typhus, or diphtheria.
The third cholera pandemic.
Yes, your answer is right. Very well done. Okay, we go to the third team, which is KMC Manipal. Yeah, can you introduce yourselves, please? Welcome.
Good afternoon, sir. I'm Akhilesh, third year medicine PG. And I'm Dr. Parth, third year medicine PG from KMC Manipal.
Right. Welcome to both of you. And I'll show you the third question.
Yep.
So scientists blame stressed out bats and habitat loss for this 21st century pandemic. COVID-19, Ebola, SARS, or D, all of the above.
C, SARS.
Okay. The answer is actually all of the above because bats were named for COVID-19. The culprit was for Ebola as well as SARS. Okay, it was implied for all three of them. Okay, right. Okay, you will get a chance to catch up. Don't worry. We go now to team number 4, which is KMC Mangalore.
Good afternoon, sir. I'm Dr. Akshay, second year medicine PG.
Yeah, and I'm Dr. Gauri, second year medicine PG.
All right, welcome to both of you. And I'll put the fourth question for you now. Doctors call her the most dangerous woman in America. An immigrant who had worked as a cook, she was New York's patient zero, passing the fever to 51 people. By the year's end, 20,000 people had died. And these are your options.
Option B, Mary Mellon.
Mary Mellon. That was very fast. Very fast response from KMC Mangalore. Well done. You get 20 points there. Okay. The next team is our very own home team, Shema. Can you please unmute and introduce yourselves to the audience?
Good afternoon, sir. This is Dr. Sanjana, my third year junior resident.
Dr. Vikram, third year junior resident.
Yeah, welcome to both of you. And your question will be put, yeah. This is the question. King Charles IV of Spain launched history's first global immunization campaign. Pathogens from a disease cow passed arm to arm in a human chain all the way from Canary Islands to the Philippines. The fight was initiated against which disease? And these are your options.
Smallpox.
Smallpox? Okay, that's right. Again, another lightning response from the team from Shema. Well done. Last, we have the team from AJ. Yeah, can you introduce yourselves, please?
Good afternoon, sir. I am Dr. Rohan from AJ, second year medicine PG. I'm Dr. Sangam from AJ, second year medicine PG.
Okay, welcome to both of you. And this is your question. Missionaries, it was alleged, had treated this virus with strychnine. A, dengue, B, measles, C, malaria, and D, smallpox.
Option D, smallpox.
Okay, the answer is actually measles. So in the 1780s, the priests were actually giving strychnine to treat measles. Okay. And it was a bit of controversy there because it was something which caused the patients to become worse and die. Okay. Yeah. So anyway, the second round of questions. Okay. Question number 7 goes to team 1 from KVG. Team 1, are you there?
Yes, sir.
Yeah. Great viral clouds of mosquitoes in World War II beat back American forces. The commanders ordered that no soldier could be excused from combat without a temperature of at least 103 degrees Fahrenheit. Which disease or which diseases were being referred to here?
Sir, C. encephalitis.
Okay, the answer is all of the above. Okay, all the diseases which are mosquito borne were implicated here. Okay, so that's what we are referring. That's the answer we are expecting. It's okay. You get chances again. Okay. Team from Yenapoya?
Yes, sir.
Yeah. This is your question. Called the romantic disease, this affliction was hailed in Victorian times as poetic inspiration. Franz Kafka, John Keats, Harriet Beecher, Edgar Allan Poe, and Fyodor Dostoevsky all romanticized the disease. Victor Hugo had it cripple the spine of Notre Dame's famous hunchback. Which disease are we referring to here?
Tuberculosis.
Yes, correct answer. Okay, we are referring to pot spine or TB. Okay, very well done. Next is KMC Manipal. Are you ready?
Yes, sir.
Yes. This epidemic corresponded with the rise of political editorial cartooning. Doctors sued cartoonists for slander, denouncing atrocious falsehoods. Which disease are we referring to over here?
B, sir. Ottoman plague.
Ottoman plague. The answer is cholera, London cholera. So basically they were sued. The cartoonist said that the cholera is just a myth, and it is the same what you are experiencing now. That COVID is not real. The same situation happened even in 1831, where they actually said that cholera is a disease created by doctors to inspire fear in people. Okay, right. Yeah. So can I have KMC Mangalore?
Yes.
Yeah. This is your question. A carved stone from the time of the pharaohs is thought to depict a victim of this neuromuscular virus. And these are your options. You have the picture for your reference.
Option D, polio.
Oh, that is a lightning fast answer. All right. Correct answer anyway. Okay, good. Well done. Well done. Okay. We'll now go to the team from Shema. Shema, are you there?
Yes.
Yeah. We are fighting a disease, not people, said Surgeon General Everett Koop in a conference on 22nd October 86. Americans would have to learn not to scorn a population of victims not exactly considered mainstream. And these are your options.
HIV/AIDS.
Yeah, exactly. That's the right answer. So Everett Koop was one of the first people to actually speak against the stigma, social stigma that is AIDS. Okay, and in fact, there was a big, he was in Ronald Reagan's parliament. And so against the current trend of people isolating or stigmatized against patients with AIDS, he was one person to speak as people should accept them into society and not isolate them. Okay, can we have the team from AJ?
Yes, sir.
Okay, this is the question. Last question for this round goes to team from AJ. When was the first influenza vaccine developed? And these are your options.
1938, sir. Option B.
Yes, your answer is right. 1938 is the right answer. Okay, at the end of round number 1, let's have the scores. Okay, we have 3 people, 3 toppers. Okay, so we have Yenapoya, KMC Mangalore, and Shema with 40 points. We have KVG and AJ with 20 points, and Manipal, sorry, but you are yet to open your account. Okay, right. Don't worry, there's a lot of chance where you get to pick up, you catch up. All right. So is everyone ready for round 2? So team number 1 gets the chance to choose. So KVG can choose the category first. So which category would they like to go for?
We didn't get you, sir. Sorry.
Sorry?
We didn't get you, sir. Category, we didn't get you.
Okay, okay. 1 second. I will.
Can you just put the categories in the chat box?
Yeah, I'll do that. I'll do that. 1 second. I'll just put the categories in the chat box now. Okay, I've entered it in the chat box. So team from KVG can choose their category first.
Sir, bacteriology.
Sorry?
Bacteriology.
Bacteriology, okay. Team from Yanapoya, choose which category you would like a question from. Apart from bacteriology, so bacteriology is used up.
Virology.
Virology, okay. Team from KMC Manipal.
Sir, mycology and parasitology.
Okay. KMC Mangalore.
Outbreaks in movies.
Outbreaks, okay. Shema.
Vaccines.
Vaccines.
Okay. So AJ is stuck with epidemiology.
Okay, sir. Okay, sir.
Okay. All right. So the first question is for team from KVG. Okay. So this is your question. Second. Good start. Yes. So intimate contacts of patients with suspected plague pneumonia should receive which of the following agents as chemoprophylaxis?
Sir, doxycycline.
Yes, exactly. Correct answer. Okay. Second is Yanapoya. Oh. Virology, no? Virology. That's okay. Next question is virology for team Yanapoya. All of the following is or are true except A. Influenza is caused by type A, B, or C viruses. B. Influenza A is the usual cause of epidemics. C. Minor changes in the surface antigens of influenza A occur every year. D. Antigen shift means major change in the influenza A virus has occurred. E. Burden of influenza B is mostly in adults. All are true except.
So is it B?
Actually, antigenic shift means a minor change.
Yeah.
Yeah, that's
So
right. D is the except.
D except D all are
All
true.
true.
Yeah, that's right. Now we go to mycology for KMC Manipal. Aspergillosis is recognized in tissue by the presence of A. Metachromatic granules, B. budding cells, C. pseudohyphae, D. septate hyphae.
Pseudohyphae. C.
It is septate hyphae. Okay. Yeah. Now we have vaccines. Yeah, we have vaccines for Shema. Shema, are you ready?
Yes, sir.
Yeah. Which of the following confers passive immunity? Hep B, MMR, Hep B, immunoglobulin, or infection with the measles virus?
C. Hepatitis B immunoglobulin.
Okay, very well done. That's the right answer. AJ, be ready for epidemiology.
Yes, sir.
All of the following are true about herd immunity for infectious diseases except A. It refers to group protection beyond what is afforded by the protection of immunized individuals. More likely for infections that do not have a subclinical phase. It is affected by presence and distribution of an alternate animal host. In the case of tetanus, it does not protect the individual.
Sir, B is wrong, sir.
Yeah, that is the right answer. Okay, well done, AJ.
Thank you, sir.
Yeah. Last question for KMC Mangalore. Which virus inspired the movie Contagion? And these are your options.
Option D, Nipah.
Okay, well done. Well done, KMC Mangalore. Okay. So at the end of round 2, let's have a look at the scores. Okay. Okay. Everyone's going along in the same place. Okay. So we have Yanapaya, KMC Mangalore and Shema at 70 points. We have KVG and AJ at 50 points and Manipal yet to open up. Okay, we'll go to round number 3. Round number 3 is connecting the images, the common link. So serial images. Yeah, please.
Sir, if I may, like the voice is cutting out in between.
Oh.
Voice is getting so.
Okay, we'll just maybe the fan. Okay, so we are having the connect the images down, which is you have to find out the common link between everything. Okay, so again, serial images will be shown in a sequence. There will be 2 images per slide. If you answer with the first image, you get 20 points. If you answer with the second image, you get 10 points, and the third image, you get 5. Okay, and there's no pass, there's no negative marking here. You get 15 seconds for each slide. So after 15 seconds, I will change to the next slide. Okay, right. So the first one goes to team number 1, which is KVG Sulya.
Zika.
Yes. Okay. Very fast. KVG is very fast. Okay. So this was the second slide and this was the third slide. Anyway, very good of them to get it in the first slide itself. Well done, KVG Sulya. The second set of images is for Yanapoya. 15. Next. Okay, time's up. The answer here is Lassa virus. Okay. Team from KMC Manipal.
Yeah.
Okay, your time starts now.
Doesn't count.
15.
Plagues.
Yes, well done. The answer is plague. So Manipal opening their account. Okay, and this was the last image. Okay, now team from KMC Mangalore. Okay, this is your question.
Cholera.
Yeah, exactly. Well done. So this was the dot map. And obviously the rice water stools and Vibrio cholera. Okay. Shema?
Yes, sir.
Okay. These are your slides.
COVID-19.
Yes. Well done. Okay. I was wondering if you guys would get it. Okay. So, all right. Team from AJ?
Ready, sir.
Yeah.
Malaria.
Yes. Very fast. Just 2 seconds. Okay. You just took 2 seconds to answer that question. That's really fast. Okay. All right. So we have the new vaccine which has been developed, and yeah, the Plasmodium parasite. Okay. So we'll have the scores again. Yeah. We have like KMC Mangalore and Shema with 90 points. We have KVG Sulya, Yenapoya and AJ with 70 points, and Manipal with 10. Okay, and now we have the rapid fire round. Oh, sorry. Yeah. Okay, so these are the rules for the rapid fire round once again. So each team, you get 6 questions. Okay, you have 45 seconds to answer all of them. Okay, it's not 45 seconds per question. You have to finish answering all 6 questions within 45 seconds. Each correct answer gets you 20 points. If you answer all 6, you get a bonus of 30. If you answer anything wrong, you get minus 5 points. Once you have answered a wrong answer, you can't change it to something else. Okay, please note that as well. You can, so in the middle, if you're not sure of a question and you don't want to answer it wrong, you can pass. And if there's time, you can still answer it again. Okay, right. So for the team from KVG, okay, KVG is ready?
Yes, sir. Yes, sir.
Okay, your time starts now. You can answer. You can answer.
2 to 8. So 2 to 8, sir.
Okay, correct. Yes. Second.
Sir, 17. Sir, second is 90.
Wrong. Next.
Sir, pass. Sir. Next, fourth question is false.
Wrong. Next.
Natural resistant.
Okay, your time's up.
The last question answer is false. Sorry, true, sir.
Yeah, but you had already told false, so it cannot be given again, and your time's up. Okay, you've got one question right. We'll now go to team number 2, that is from Yanapoya. Okay, answer the questions in sequence. I will not be reading them out. If you want to pass, you can pass them. Your time starts now.
Pneumococcal vaccine, Haemophilus influenza.
Okay.
March 2021.
Okay.
Epitope.
Yes.
True.
Answers false.
Carbapenem resistance. Pass.
Last one.
I don't remember.
Parvovirus B19.
Wrong. Okay. We'll tell you the scores later. Anyway, good performance by Yanapoya. Team from Manipal, are you ready?
Yes, sir.
Okay. Your time starts now.
Corona vaccine protects against 4 strains of Vibrio polyviral, but there are 4 strains. It's going to be maintained in social distancing. This is what 6 feet. 6 feet per second. Second 6 feet.
Okay.
Primary objective screen is to trends and occurrence of specific diseases. That is false. Yes, that one is false. Why? Primary objective screen is to trends and specific diseases. False. Third is false.
Okay.
Activation of T cell requires initial humoral response. That is true. No, false.
Okay, correct.
Rose spot matula rash is seen in typhoid.
Yes, correct.
First vaccine was developed against smallpox.
Yes.
Oral corridor vaccine prefers true. Yeah, true. First one.
Okay. Yes, Manipal has done a very good job answering all 6 questions right. You get the bonus points.
That's pretty easy.
Well done. Okay. KMC Mangalore. KMC Mangalore, are you ready?
Yes.
Yes. The time starts now.
First one, complete from last weekend. Beta D glucan is third one.
Third one is?
Beta D glucan.
Wrong. It's galactomannan.
This is considered father of public health. Option last one is oral polio vaccine.
Correct.
And WHO when they declared COVID-19 outbreak is pandemic in March 2020.
March 2020, COVID was declared as a pandemic.
Yes, and that with that
global
time is up. Okay, you've got 2 of them right. Okay, we'll go to team number 5, which is from Shema. Shema, are you ready?
Yes, sir.
Your time starts now.
Sixth one is false, sir.
Yes.
Quarantine was first applied for some infectivity. The time it infectivity. It's not like a short integration correct.
False.
False.
Parasitic
4 is false, sir.
4 is false, sir.
PC 23 is for elderly individuals.
Yes.
So second question, can you tell, sir?
It is not shown properly, sir. Second question.
Dash became the first state in India to conduct pool testing for COVID-19.
Kerala.
False. And with that, your time's up. We go to the last team, that is AJ.
Yes.
AJ, are you ready?
Yes, sir.
Okay. Your time starts now.
Protection commences dash to Mucormycosis oral T120. Second one, Mumbai, sir.
It's Pune.
First one, 7 days, sir.
It's 10 to 14 days.
In BCG vaccine. Word quarantine means 14 days false.
Yes, that's correct.
BCG vaccine BCG vaccine Edmundston Zagreb. Sorry. Curing.
Okay.
Third one is true.
Third one is false. And with that, your time's up. Okay, we'll just total the scores. Give us around 1 minute. A lot of changes, dynamic changes have happened in the totaling. Yeah. Yes. Okay. This we have the final scores. Okay, it took a lot of calculation and a lot of changes have happened. You won't believe the outcome. It has caused a major uproot and upheaval. So the winning team is KMC Manipal with 160 points. Congrats to all of you. You have come, you're the dark horse. You have come against all odds and won the quiz. The second team is a team from Shema with 145 points.
Where are you going?
What?
Thank you, sir.
Yeah.
Thank you, sir. Thank you, sir.
Yeah. Congrats to both of you. Okay. I'll read the other team scores. Okay, just for your knowledge. So KMC Mangalore came with 125 points. And next followed by Yenapai with 120 points. And KVG and AJ with 75 points each. Okay. Thank you all. It was a very...
Rohit, before we end, can you please give all the teams the answers for the rapid fire round? So that everybody goes back with some added knowledge.
Yes, definitely. So we'll give you the answers for all the rounds of the rapid fire. So team 1, as you all told, the answer, maintaining cold chain ensures vaccines are stored according to manufacturer instructions at 2 to 8 degrees. That is right. Minimum percentage of alcohol to be present in hand sanitizer to offer adequate protection against COVID is 60. This answer is from the WHO guidelines. Ability of an infectious disease agent to cause severe disease is called virulence. Candida krusei is naturally resistant to azoles. The answer is true. Other name for Catayano fever is schistosomiasis. 2 live vaccines cannot be given together? The answer is true. It can be given 4 weeks apart.
That alcohol percentage, can you repeat, sir? How much?
60%, 60. This is in the CDC guidelines as well as the WHO. Okay.
Thank you.
Yeah. Team number 2. The answers are vaccinations for splenectomized patient, as you have said, Hib, pneumo, and also meningococcal. The second phase of COVID vaccine began on March 1st, 2021, which you answered. The subunit of antigen which binds with a specific immune receptor is called epitope, which is right. The Diziafulvin is used to treat systemic fungal infections. The answer was false. It is used for topical. Carbapenem resistance is due to mutation called NDM1. Nitte disease is called tetanus neonatorum. Answers for team number 3. Oral cholera vaccine protects against 4 strains of Vibrio cholera O1, which was true. Minimum advised distance to be maintained is 6 feet or 2 meters. You are right. The primary objective of screening is to establish trends in the occurrence of specific diseases. The answer was false, rightly said by you. Activation of T cell requires an initial humoral response. The answer is false, which you answered correctly. Rose spot macular rash is seen in typhoid, which is right. The first vaccine was developed against smallpox. The answers for team number 4. Those immunized with H influenza B who then develop splenic dysfunction need no further doses of HIV containing vaccine. The answer was false. WHO on March 11th, 2020 declared the COVID outbreak as a pandemic. We were right. The most specific test for diagnosing invasive aspergillosis is galactomannan. The first line antibiotic in the treatment of Shigella dysentery is ciprofloxacin. Disease considered as the father of public health is cholera. And the reason being the first dot map way of tracking a disease was done for cholera. The vaccine associated paralysis is more with OPV than IPV, which you answered correctly. Team 5, PCV23 is recommended for elderly age group. It's age more than 65. Uttar Pradesh became the first state in India to conduct pool testing for COVID-19, where you take samples from 5 individuals and test them for PCR positivity. If it's positive, these patients are then tested individually. The sample turns positive, you again do a PCR for those 5 individuals. Okay, it saves time and saves cost too. Time between infection and maximum infectivity is called generation time. Parasitic infections have a long incubation period. So the answer here was false, and you have answered correctly. Quarantine was first applied for plague. So the first disease was plague. WHO Health Day is April 7th. Okay. So your answer was correct here when you said false. Team number 6. So protection commences 10 to 14 days after complete primary course of oral type 21A vaccine. Reliance Industries set up the first COVID dedicated hospital in Pune. NK cells do not need to be induced or primed to kill agent-bearing cells. The answer is false. The drug of choice used in treatment of anthrax is penicillin. The word quarantine means 14 days. The answer was false, which you answered correctly. It means 40. Quarant means 40. And the strain use of BCG is Danish 1331. Okay, so with that, the answers, this concludes our quiz session. I thank all the participants for attending and making this quiz a success, and congrats to the winners. Others, don't be disheartened, you have gone wiser. Thank you all for participating.
Thank you, sir.
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 Thank you.
Thank you, sir.
Yeah, thank you.
I can do this.